I got you a mic. I didn't, because are they going to ask questions?
Yeah.
Remember we did that?
Do we run the mic up?
It's on. It has just has to be turned. Only if you want to. It's up to you.
Good afternoon, everybody. Please come on down to the front rows. We're going to move everybody out of the back 6 rows, please, up to the front 6 rows. We want you to be here. If you need to sneak out, you could sneak out the front. So let's try to fill in a little more up to the front, please, for all the people coming in. It'd be great. Feels kind of funny with half a room in the back. Come on, guys. Don't be shy. We won't call on you. Thank you. Thank you. Thank you. Thank you very much. Poor Dr. Vaughn is sitting alone in the front row. Okay, thank you very much for helping us out with distributing the seating. So I think we should get started. Welcome, everybody. This is always a great day every year at the School of Medicine, and this year is no different. It is one of the highlights for the program, and hopefully for all of you students. It certainly is for the faculty and the leadership of the institution, in that we are extremely proud of the work that the medical students have done over their 4 years in terms of the research, along with the other value domains as well. And this is a day to highlight and pay tribute to the research that you all have done. So we're very excited to hear from the speakers today. Let me just say, by way of background, we're not only grateful to the students and all the work they've done, but also to the many mentors that you've worked with. So if you wouldn't mind joining me in a round of applause for all the mentors that have worked with you. And speaking of mentors, it's become a tradition of rather short order, but one that we will continue well into the future, to recognize the mentors as well, and in particular to select an outstanding mentor, or have you select an outstanding mentor and recognize them before we get into the presentations by the students. So it is my great pleasure today to tell you a little bit about the process for that selection, and then to introduce the person who was selected for this recognition this year. So firstly, as I think most of you know, the nominations for Outstanding Mentor go out to all of you, all of the medical students, not only those of you here now, but to the graduates who have come before you as well, who have worked in many cases with these mentors. They are asked to evaluate and use a series of criteria for selection to recognize them. I'll just remind everybody and tell you it's for creating an environment in which original hypothesis-driven research is effectively implemented and guided, to provide opportunities for your professional growth, to challenge the mentees to make original research contributions and carry your research to completion with rigor, to foster intellectual growth and technical and methodological proficiency, and to establish clear expectations and maintaining high standards for quality, research ethics, and work ethic. And so that's a pretty high bar and a lot of things that we expect, and we got some really terrific feedback from many of you and many of the earlier graduates as well. And it wasn't an easy decision. There are a lot of terrific mentors, and you know that, I think, but the group of people on the committee were able to come to a consensus and recognize the outstanding mentor this year. And that person is Dr. Stephanie DeLuca, who is sitting right here. Please join me in prayer. And let me tell you a little bit about what you said and what your colleagues said about Dr. DeLuca as a mentor. First of all, she has mentored 6 medical students so far, and I have an example of several of the letters that the students wrote about her, and I'll just read you a couple excerpts from that. One student said, "I told her my simple questions and ideas, and she helped me to create a project of my own that delved into my research interests while also being able to work with the patient population. I'm so grateful she gave me the time to explore my interests and engage my curiosity, as well as for believing in my project ideas and helping me to create a feasible hypothesis." Another student said, "My appreciation for the research process grew, but I also became a stronger researcher as a result of working with Dr. DeLuca. I began to think not only about what my project sought to accomplish, but how it fit with the larger picture of the field. What new questions arose from the one I tried to answer, and what limitations still existed in the field. I also became a stronger writer and better able to disseminate my findings succinctly and effectively in the process." And one other student said, "She also helped me to take ownership of my project using resources effectively, but always challenging me to not only complete the tasks for my project, but also to understand the methodology of my project from start to finish. The skills I honed in study design, biostatistics, presentation, poster preparation, grant writing, manuscript writing will be indispensable for me as a busy resident and attending in the future." So I think these capture the essence of what many of us have known about Dr. DeLuca for a long time. I've had the pleasure of working with her for many years at another institution in here, and I've seen this mentorship and passion and dedication she has for students, and I'm so glad to see that many of you recognize that as well. So based on that, we would like to give Dr. DeLuca a plaque to recognize her and have her come up, and then she will have some time to give us a talk. Somewhere there's a plaque. They're pointing. Behind you. So Dr. DeLuca has some time to give us a little bit of a talk about her own experiences. Stephanie, welcome.
Wow. First, let me say this is an incredible honor, and I'm very appreciative to everyone and to all my current and previous students, and I cannot tell you how thrilled I was to receive this award. I'm not going to actually talk too much about my previous work. I mean, this is what I do. I put up a bunch of pictures. I work with kids. I try to help kids with challenges and create skills and gain function. Now, I don't do this alone, but this is why I do what I do. All of these smiling faces and children working hard is what keeps me going, is what my dedication is based on. Now, I think society holds a lot of misconceptions about children with disabilities. And I think that many of these pictures indicate some of the things that they can accomplish, because I can tell you a lot of these children were thought that they wouldn't be able to do some of the things you see them doing, whether it be as simple as trying to crawl or scooping beans or holding an object. We often tell children with disabilities and families with disabilities about things that they can't do. And I've spent my career trying to find out what they actually can do. Now let me see if I can make this go in the right direction. Most of my work has surrounded intensive therapies, particularly constraint-induced movement therapy, which says that we need to give children intensive bursts of therapy. Most of the studies I've done have been focused on children with hemiparesis, and I have come up, or my group has come up, and I say group because it's really not just me, has come up with a specific form of intensive therapy, pediatric constraint-induced movement therapy, called acquire seed therapy. I think one of the biggest things that we've done for the field is try to operationalize what therapy should be. Rehabilitation in its history doesn't have a strong consensus about how to treat children with any type of disability. It's been a hand-me-down approach, and operationalizing protocols has not been something that's consistently done. So if you ask me about what I think the biggest contribution to our field, it isn't about a specific protocol. It's about trying to encourage people to operationalize treatment protocols so that they can have the broadest impact across children with disabilities. Now, in this process, I've been involved with a lot of clinical trials. This is just a few, and this is the only data point here that shows traditional therapy. And I hope what this shows you is that across the clinical trials that we've done, we consistently get pre to post changes, if I can go back, sorry. We consistently get pre to post changes, with pre-treatment being blue and orange being post, across many, many different trials, but there's not much difference in controls. So these are children that are getting traditional therapies. And it's been an honor to be a part of all of these trials and upcoming trials. But I think the one thing that we've learned is that intensive therapy, given in bursts of treatment, work for children. They work for children in helping them learn and gain skills. Now, I have a lot of different interests, and I want to take this a lot of different directions. And the students are the group that helped me do this. I want to take this idea of intensive therapy to other etiologies, such as microcephaly, to lower functioning children who have greater impairments than just hemiparesis. I've had the pleasure of doing health disparities work internationally, so I want to continue that work. And I also, of course, am here at this institute and in this School of Medicine because I have a strong interest in understanding the impact and changes that intensive therapy has on the brain. We see the changes functionally, but thus far we really haven't been able to document the changes that intensive therapies have consistently on the brain. And I want to understand that with future studies. But this is really about students. So I think students should be involved in all of those things, and I want to involve them in all of those aspects. But I wanted to talk with you briefly about what mentorship is. So I'm going to tell you some things that's not shocking. I learned mentorship. Many of you know that my primary mentor is Dr. Sharon Ramey, and she's here at this institute with me. I now consider her a colleague. But she was my mentor, and I learned much of what I do with students from her, and so I need to acknowledge that. Sharon, along with others, including Mike, who I also considered a mentor, challenged me. Okay? I learned mentorship through being challenged. They also listened to me, which I think is key. Okay? It's not enough just to say what a student should do. You have to listen to their ideas, and you have to listen to their interest and what they want to learn from and learn about, okay? But a major thing that I also think is important is challenging students to not accept the status quo. That's what research is really about, but that's also what medicine is about. Medicine is driven forward by trying to make changes, by trying to challenge what the current system is. Now, mentorship is, of course, about passing on the baton, okay? The next group of leaders, the next group of scientists, the next group of clinicians will have to develop research projects. And I think that the need for scientifically valid clinical research is increasing. It's not decreasing. So we, in our current status as scientists and clinicians, have an obligation to make sure that students are trained well. And I also think that it's important for all clinicians to be versed in science, But clinician scientists are truly what lead the way, and what lead the way in changing the system. And that's why I think I'm so blessed to be a part of this institution, because I know that many of you are not necessarily thinking about being a clinician scientist, but the curriculum here is really driving that point home and at least preparing you for that consideration. All right? But for me, mentorship, I must also acknowledge, has always been about changing the system. Specifically for me, the rehabilitation system. I hate to report this always, but it's a striking idea. In 2013, there was a review that shook up the system, and they actually looked at almost all rehabilitation treatments currently used in pediatric rehabilitation. And they judged only 16 interventions worthy of recommending. That's pretty striking. That's not just therapies. That's not 16 therapies. That means if you throw drug products in there, if you do therapies, if you do surgeries, only 16 of everything we do for children with developmental disabilities they could find have efficacy. Worse yet, they judged that 20% of the treatments currently in use, that are currently prescribed pediatric rehabilitation treatments, might be harmful or at the very least have iatrogenic effects. To me, those numbers must change. I mean, think about that. We are prescribing things. Children are receiving things that the current literature suggests might be harmful. So I think that's a problem for mentorship. I think mentorship is about creating leaders. It's about guiding, creating leaders that can change the system. All right? Clinical care should be based on sound scientific findings. But to do that, clinicians need to understand and be able to interpret research. And there's no better teacher in doing that than experience. I bet all of you know more about research than you ever thought possible when you entered medical school at this point, whether you're a first year or a fourth year. My guess is this particular institution, because of its focus on research, has taught you things that you didn't expect to learn, and I applaud that. Now, I also expect my students to take all of this and challenge the healthcare system in their future careers. That's important to me, and I point that out to my students. I want them to learn many different things, but I want them to have their research curriculum to have a lasting impact, no matter what direction they go, whether it's rehabilitation, whether it's surgery, whether it's oncology or neurology. I think the lessons that they learn in research are applicable, because pediatric rehab is not alone. There are many things across the medical spectrum that we currently do that need to be changed. Good mentorship is also, as I've already kind of alluded to, sometimes occurs through great environments. And so I have to acknowledge the School of Medicine and the Fralin Biomedical Research Institute as creating that great environment. The encouragement for interaction with students, the encouragement for interprofessionalism is not duplicated at most other institutions. So you should be thrilled that you are gaining this early in your careers, because this is, again, something I think that's going to become more important, not less. I also have to acknowledge that good mentors come because I have great students, okay? I am very appreciative of all of my students, some of which you're going to hear from, but I at least wanted to take the opportunity, in no particular order, I need to say, to introduce you to the students that I currently have, okay? Vaishnavi is actually working on fMRIs to look at neurological changes. She has an interest in mirror feedback and mirror neurons. So she's looking at individuals who are typical and individuals who have neuromotor impairment and trying to understand the use of, or if the mirror neuron system, how much it is in use with actions and that are actually completed. Omar, you will hear from shortly. He has used the Xbox Connect system to try to develop a better system for evaluating children who have neuromotor impairments, and he too has looked at typical individuals and individuals with neuromotor impairments. Ken has looked at is developing the psychometrics of the emerging behavior scale. This is something most of you have not heard about, but when I started working in pediatric rehabilitation, one of the things that plagued the system before and still plagues the system is we don't have good measures to measure change in children. And so the emerging behavior scale is actually something that we developed early on in my work, and I was very surprised that it got picked up by a lot of people and has been used in numerous studies right now. But it doesn't have the psychometrics yet established. And so Ken is working on trying to develop the psychometrics and establish them. And then Miranda is interested in using constraint-induced movement therapy and training individuals in low-resource countries. And she recently traveled, or a few months back, traveled with me to Vietnam and is looking at how the training processes for therapists in Vietnam compare to the training processes to therapists here. Lastly, I want to say that mentorship, in my lab at least, is never done by a single individual. These are two pictures. I couldn't decide on which. There's some duplicative people here, including some students. But all of these people are people that work in my lab and that are contributory to the process. Students interact with lots of different types of professions. They interact with with occupational therapists, psychologists. They also interact with educational people across the board, and that's very important. And I need to say that I didn't mentor any of these students, past or present, without the help of these individuals. And with that, I thank you all.
Okay, thank you very much, Dr. DeLuca. We are going to now get started with our presentations from students who've earned letters of distinction in research scholarship. So these projects were chosen by the evaluators who come in and listen to your fourth-year oral presentations. And those evaluators say, yes, no, nominate, don't nominate, and then a committee gets together and looks at the projects and decides based on overall quality, significance, relevance, innovation, and clarity of the students' findings, and looking at both your written form and your oral form. So we've picked the top 8 students from the class of 2019. It wasn't an easy choice. This is a great class full of very accomplished researchers, but we're going to hear oral presentations from them. The presentations are going to be 15 minutes, and we're going to have time for maybe 1 or 2 questions between each student presentation. And the first presenter we're going to hear from actually worked with Dr. DeLuca. So Omar worked on a project entitled Evaluating Connect Sensors as a tool to accurately measure and monitor progress of occupational therapy in pediatric rehabilitation patients. Omar.
All right, hi everyone. Dr. LaConte just told you my title, so I won't repeat it because it's quite long. But my name is Omar, and I work with Dr. DeLuca, and I also want to recognize Dr. Alan Asbeck at Virginia Tech's Department of Mechanical Engineering. So he's also instrumental to my project. So, quick background. So most children with neuromotor impairment, as Dr. DeLuca alluded to, are referred for occupational physical therapy. But the majority of these therapies cannot actually document clinical efficacy. And it's thought that this is possibly due to the inability to accurately and objectively assess patient progress. So there are a lot of subjective functional assessments that are often used in clinical practice, but they don't actually provide any objective measurement of like a joint angle or an actual number that you can use to track over time. And so it's thought that an objective and quantitative measurement of a physiological characteristic would enhance the understanding of rehabilitation outcomes. And so to frame the study further, the project motivation and the physiological characteristic that we chose was active range of motion. And so active range of motion simply means that it's an activity that you initiate yourself as the person who's doing the activity. So you can't have someone move your arm for you, you move your arm, and then the joint angle is measured, and then that is able to be tracked over time. And it's been tied to functional capabilities of the arm and the hand, and our study focused on the upper extremity. And so the gap in knowledge that we were trying to address is if accurate and objective measurements of active range of motion can be obtained to improve processes and outcomes in pediatric rehabilitation. So if having this number would be valuable in actually tracking the progress of these patients. And so the research question was if the Xbox Kinect could accurately measure joint angle in the upper extremity to provide an objective measurement of active range of motion. And so why the Kinect specifically? So in short, teens. So how many of you have been teenagers? Should be everyone. And how many of you have played video games? Okay. So actually, studies show that up to 97% of adolescents play video games. And the issue is that specifically once people enter adolescence, often the rehabilitation resources are particularly limited. And so for example, at our research clinic, some of the patients who I interacted with came from Canada just to do therapy at our center. And there are very few centers that actually specifically can function at the level of the pediatric requirements. And so if you add a teenager and possibly add a Kinect, then you won't get a grumpy teenager, you'll have a happy teenager. And the reason for that is because the Kinect would be able to do the things that right now are being done by an occupational therapist and require someone to actually go to one of those specific sites to actually receive the therapy and possibly do that therapy at home. And not only do the therapy at home, but possibly do it in a video game, which teenagers are already using. So further information. So the home-based therapy has actually been shown to be equally effective and less costly compared to center-based therapy. And also, active video gaming and motion capture systems have been thought of as a potential gateway experience for neurorehabilitation. So there are already certain centers that are using virtual reality and things like that in their hospitals for rehabilitation. However, it's important to note that these are not really evidence-based at this point. So not having a specific outcome like active range of motion or a physiological characteristic limits the interpretation of the data and if the children actually improve from participating in these therapies and creating a standard and operationalizing, as Dr. DeLuca alluded to. And so the implementation right now is also limited due to cost, accessibility, and lack of specific outcome measures. So right now there are sort of better systems like the Vicon system, which is a motion capture technology where you wear markers and then it kind of is able to track in real time a kinematic analysis of how the subject is doing. That can cost upwards of $70,000. You can buy a Kinect for $50 on eBay. So you can imagine that if this is something that's beneficial, it's also cheap and something that children might already have in the home. And so the ideal rehabilitation tool would be accurate and reliable, accessible and affordable, and effective and measurable. So we kind of know that hopefully it's accessible and affordable compared to the current systems. And so really we're targeting to see if it's accurate and reliable in this study. And so the current tool is a goniometer. So this is a goniometer. It kind of looks like a fancy protractor. Essentially how it works right now in therapy is the occupational therapist would go up to each child while they're doing the therapy and measure their joint angle, like put it up against their limb while they're doing this activity. So as you can see and imagine, this would be very time consuming. But what's important is that the goniometer, though it's a little cumbersome to actually use in practice, it has been shown to be the clinical standard and to be accurate in measuring the joint angle. And so that's sort of the clinical standard that we were comparing it to. And the goal of our study was to show that the Kinect was able to be equivalent to the goniometer measurements. So our hypothesis, again, is that the Kinect sensor would provide accurate and reliable measurements of active range of motion compared to goniometry. But I did want to give you a quick overview of how the Kinect works. I'm not a complete expert, but essentially it has three sensors. So it has an infrared projector, an infrared sensor, and an additional sensor that's able to actually triangulate the joint angle. And so here what it does is it projects an infrared dots onto the body that's in front of it. It obtains a depth image, and from there it uses all the different random series of dots to assume this is the body part distribution. It's able to tell generally this is the upper limb, this is your arm, because it can kind of tell what part is the trunk and what part is the limbs. And then from there, this is happening in 3 planes. And so because it's happening in 3D, as you can imagine, you have sort of 2 points where you're able to say this is a limb, and you also have 3 different planes because it's an infrared sensor. So what we're able to do is actually create Cartesian coordinates, and that's important because we can actually, although the Kinect system is looking at the entire body, we're able to focus on a specific joint and a specific motion. Because as you can tell, when you're moving your wrist in multiple directions, what we might be interested in when we're looking at the study is specifically flexion or extension or radial or ulnar deviation. So this helped us to be able to actually isolate each specific joint angle that we were interested in measuring as we were going. So methods in terms of our subject enrollment. So we started with typically developing subjects. We had 64 who were typically developing, and we had 32 who had neuromotor impairments. So we tested 96 people overall. And 38 were female, and 26 were male in the typically developing group, and 14 were female, and 18 were male neuromotor impairment group. And we tested both adults and children, but we did end up looking at children separately in some of our analyses, but our median age was about 18 or 19, and our range was about 5 to 76 total. We didn't look at gender, age, or ethnicity, or weight in terms of exclusion criteria because really all you needed to do to participate in our study was to be able to move your arms. And it didn't really matter specifically if you were able to achieve maximum range of motion because all we were looking at is if the number between the Kinect and the goniometer was the same, it didn't have to be 90 degrees or a specific angle that we were looking for. And just so you're aware, the diagnoses in the impairment group were cerebral palsy. 18 of our patients had that. 10 had a stroke, 2 had traumatic brain injury, and 2 had a spinal cord injury. So in terms of our data collection, we got consent because that's good to do. We also set up the Kinect sensor system and connected the laptop to obtain the output in real time. And then as I mentioned before, they participated and performed all the active range of motion activities at the shoulder, elbow, and wrist in both the right and left extremity in front of the sensor, and we collected both separately. The joint angle was measured simultaneously by the Kinect system and also by the occupational therapist using the goniometer. And then we compared the goniometer data with the Kinect data at the same exact timestamp so that we were able to see if the numbers were equivalent. So this is just a quick picture of our setup. This is where the subject is standing. The Kinect would be here. And these parameters are just based on the optimum accuracy that's been shown in previous studies for the Kinect sensor system. So just to give you a quick view of what it looks like. So this is what you would actually see if you were using the Kinect. And so what you can see here is the circles are, well, the big one is the head. And then the smaller ones are each of the joints when you're looking. And so you can kind of see that everyone's moving their arm. And so we chose these 14 different activities because they're actually tied to functional rehabilitation goals. So things that we don't really think about because most of us are typically developing is that things like radial and ulnar deviation, they seem normal to us and it's kind of like, why do you test it? But the reason why is, I mean, just imagine it's important if you want to open a jar. Having internal rotation is important if you want to put your hand behind your pocket. If you want to open a door, these are things that we kind of take for granted, but they're actually very closely linked to the function that would be of interest in someone who has impairment. And so our results, so our aim, as I mentioned before, was to assess agreement. This is an equivalence study. And so the agreement that we defined it as being the difference in measurements within a clinically acceptable margin of error. The number that I want you to remember is plus or minus 5 degrees. So it's kind of our magic number that's been shown in the literature to be the acceptable margin of error between the measurements of the Kinect and the goniometer. The analyses that we used were Pearson's correlations, non-parametric analyses, Bland-Altman plots, and paired t-tests. I'm going to share just the highlights with you during the presentation. So I know that M1's just had their biostats exam. So this is shoulder, and we're looking at a correlation. So essentially, we looked at correlations because even though correlations show that everything is sort of in alignment, it doesn't necessarily show that there's agreement. So we did this just to have a global idea, because if you can imagine, 2 numbers can be very strongly correlated, but that doesn't mean that they're near the same number. So we did this at the shoulder, the elbow, and the wrist, and as you can see, they're all straight lines, very positive, and had strong correlations, like here you look at 0.989. So that was sort of a global picture, told us that we were in the right direction. We also sort of looked, as I mentioned before, our sort of magic number was 5 degrees. So here we actually looked at the number of measurements, the number of times that the difference in the measurements was greater than 5 degrees versus less than 5 degrees. And this is the typically developing group, and this is the neuromotor impairment group. What you can see here is looking at the shoulder, the elbow, and the wrist. When you take everything together, only 8% of the time in the typically developing group there was a difference of greater than 5, and only 1% of the time in the neuromotor impairment group was the difference greater than 5 degrees. So that to us said that we sort of, yay, we achieved the plus or minus 5 degrees that we wanted to achieve. We wanted to keep looking at it. So what you see here is average absolute difference. And so the reason why we did that is because, as I mentioned before, we're looking to see if the measurements are the same, but the difference can actually be in the positive or the negative direction. So we looked at everything and made it positive, and then this is looking at every specific movement that we tested in the shoulder. And what you can see is within all of these different movements, it fit within one standard deviation of the plus or minus 5 degrees that we were looking for on average for all of the movements. What we also see here is that the neuromotor impairment group, like before, had stronger general agreement than the typically developing group, and we'll talk a little bit about that later. But then again, at the elbow and again at the wrist, we see the same exact thing, where everything is very strongly showing that it is within the plus or minus 5 degrees that we were looking for. So this one's a little bit more of a doozy, but it's a Bland-Altman plot. So essentially what we're looking at is here, this is the average between the connect and the goniometer, and here's the difference between the connect and the goniometer. And this is for a specific movement. So here we're looking at radial deviation in the typically developing group and the neuromotor impairment group. So what we really need to take away from this is that we want everything to be 0, because that would say that the measurements all were 0 in terms of the difference. They were exactly the same. We didn't get quite 0, but more importantly, we also wanted everything to be between the plus or minus 5 degrees. And again, we sort of see the trend that here it's plus or minus 5 degrees, but the neuromotor impairment group is plus 2.38 and minus 3.88. So again, it's sort of more tightly agreeing than the typically developing group. And this was true for every single movement that we looked at, all 16 measurements in both the right and the left limb. So we also did some additional analyses. So as I mentioned before, we did the correlations, and those are for all of the joints across all movements, but for every specific joint movement that we had, there was strong correlation between the connect and the goniometer within plus or minus 5 degrees. We also did paired t-tests, basically just to see if there was any significant interaction between if we were looking at impaired versus unimpaired subjects, if we were looking at child versus adult subjects, to see if maybe it's better at picking up adults because they're bigger, and so the depth sensor would work better than in children. We didn't see any significant interaction for any of these things that we were looking at. And then finally, we were also looking and did a linear regression analysis, which showed no fixed or proportional bias in the measurements. And essentially what that means is the connect wasn't always off by 2 degrees, and the connect also wasn't suddenly bad at getting the data when you were at a certain angle that was either too high or too low. So our conclusions were that the connect sensor system is a valid tool to measure joint angle in the upper extremity during active range of motion, and it does have potential for use in the rehabilitation setting, and also could be integrated with additional technologies to participate in rehabilitation. Some observations, limitations. Something that was interesting that I wasn't really expecting with our study was that there was stronger correlation, greater agreement with the neuromotor impairment group, which would seem kind of confusing at first because you would think that they have less range of motion. But the more we thought about it, we realized that it might make sense because these children are constantly going to therapy. They're constantly being sort of poked and prodded and having everything measured. And so they probably know exactly what to do when they're seeing a rehabilitation therapist. And also, something else to think about is that when you have less range of motion, inherently the differences are going to be smaller because you have less to sort of deviate from. We also had fewer impaired subjects than typical subjects. That's just another significant limitation of the study. We also did not repeat measurements in the same subject or compare these to functional assessments. So I alluded at the very beginning to the subjective measurements that we use to look at therapy, but right now we didn't specifically look at those during our study. Another observation, the clinical standard of plus or minus 5 degrees was great because it was something that we had in literature that we could refer to. However, specifically at the wrist, that's a fairly significant angle or difference at the wrist. So I think for larger joints like the shoulder and the elbow, it makes sense, and that's more clinically acceptable. But to think of a child with neuromotor impairment, they might not even have 5 degrees at the wrist. So in future studies, we might consider having our own sort of standard that we create for the wrist in the future. And also, we had the ability to look at everything in real time, but we chose to actually specifically have the child sort of do a freeze frame and then look at their joint angle in that one moment. But actually looking at a real-time kinematic analysis could better elucidate some of the therapies and the efficacy of the therapy. So future directions would be to compare this with the functional assessments and motion capture tools that I mentioned before, also to explore virtual reality or Connect as an actual therapeutic approach as opposed to just monitoring progress over time and doing the therapy in the traditional sense. So I wanted to go ahead and recognize the team, Dr. DeLuca, who you just heard from, Dr. Asbeck, and then also our amazing senior occupational therapists, Mary Rebecca Trucks and Dory Wallace, and Laura Bateman, the senior administrative assistant. Dr. DeLuca has been one of the most influential people in my medical school career and has really informed my decision to go into academic pediatrics, and so I'm really thankful for her for helping me and believing in my project and aiding me throughout the last, maybe sometimes more than other times, aiding and helping and doing things over the last 4 years. She's been really amazing. And also, obviously, thank you to all of the participants who participated in my study. It was really meaningful, and interacting with these families has really been one of the highlights of my medical school career. Thank you.
So we have time for one brief question. If anybody has a question to ask Omar.
Thank you for that excellent presentation, Dr. Solomon. Just a follow-up question to your presentation. Do you think your research could be used as a tool to help us better identify when conservative therapies or modern Medicaid patients it's time to pivot to surgical intervention?
So that's an interesting question. I think that in general, choosing to do surgery depends on the patient, and also something that we didn't specifically mention with our study is that we looked more often at children who had very limited impairment. So most of these children, as Dr. Luca mentioned in her talk, have hemiparesis, or most of the adult subjects had very mild impairment, and so they were very easily able to perform and participate in the therapy. I think if you were to use this in someone who is more likely to get surgery, which is someone who has quadriplegia or more significant impairment, then I think that that would have to be validated in that specific patient population. So I think we sort of self-selected for a group that probably would be more likely using conservative measures like occupational therapy as opposed to having surgery.
Thank you.
Thank you.
So our next presenter is going to be Gail Stanton. Gail worked with Dr. Michael Fox at the Fralin Biomedical Research Institute, and she's going to talk about the role of activity-dependent gene NRN1 in retinogeniculate synapse maturation and visual behavior.
Hi everyone, thanks for coming to my talk today. My name's Gail and I'm going to be talking about our work that we've been doing to study the development of retinogeniculate synapses, or the connections that form between the eye and the brain. In the Fox Lab, we study the nervous system. The nervous system is made of neurons, which are highly specialized cells that carry information. Neurons are made of dendrites, which receive information, and axons, which send information out. The nervous system is made of billions of neurons, and many of them are highly specialized to perform specific functions. In the Fox Lab, we specifically studied the visual system. As humans, we're highly visual creatures. We use vision to interact with our environment, to identify hazards, as well as to pick up on social cues. And how do we do this? The visual system is a highly complex system, and this begins with special cells in our eye that actually detect light. This information is then sent back to the brain by cells called retinal ganglion cells. Retinal ganglion cells send this information back to the lateral geniculate nucleus in the thalamus, which acts as a relay center. And from the lateral geniculate nucleus, this information is sent back to the cortex, where it is processed and used. Now, the lateral geniculate nucleus is actually composed of several main parts. The ventral lateral geniculate nucleus, or the VLGN, receives visual information from the eye that's used for non-image forming purposes, such as for circadian rhythm. And the dorsal lateral geniculate nucleus, or the DLGN, receives visual information from the eye that's used to form images in the cortex. Historically, it's thought that information travels through the system in parallel pathways, such that the layout of the different parts of visual information is preserved and then can be reassembled in the cortex. However, in the Fox Lab, we recently discovered something very interesting when we compared synapses in the VLGN and the DLGN. When we reconstructed synapses in the VLGN, like you see here, we found that synapses are formed from an axon from one retinal ganglion cell and a dendrite from one thalamic cell. In contrast, when we reconstructed synapses in the DLGN up here, we found that these synapses are formed from axons from many different retinal ganglion cells that are converging onto a single thalamic cell. This means that unlike the layout I just described to you on the previous slide, there's actually a high level of convergence in the visual system, and information is not traveling in those parallel pathways like we thought. And this was completely new when we discovered this. So one of the first questions we asked was, how do these synapses form in the DLGN but not the VLGN? So in order to answer this question, we looked and found a number of proteins that are highly enriched in the DLGN when these synapses are forming. And therefore, we looked at them as candidate genes to be involved in this process. Of these candidate proteins, we picked two, LRRTM1 and NRN1 is our candidates to study. LRRTM1 is a protein that has both extracellular and intracellular components, and it had a known role in inducing excitatory synaptogenesis. NRN1, or neuritin-1, is a protein that's secreted and membrane associated, and it had a known role in neuritogenesis and dendrite arborization. And additionally, there was a previous study that demonstrated mice lacking NRN1 actually had delayed growth of the area of retinal projections to the DLGN. So based on their structure with those extracellular components and the previous studies indicating their roles in synaptogenesis and neuritogenesis, we thought these two proteins were kind of our strongest candidates to be involved in the development of these complex synapses. However, interestingly, when we studied mice lacking NRN1, we actually didn't find any differences in those complex retinogeniculate synapses. And additionally, we weren't able to replicate that previous study showing that mice lacking NRN1 had that delayed growth of retinal projections to the DLGN. So that kind of left us with questions of, if NRN1, we didn't find any evidence that's involved in the development of these synapses, what is its role in the DLGN and why is it enriched during development? In contrast, when we looked at mice lacking LRRTM1, we actually found that they lost those complex synapses. You can see this quantified in the graph on the left here. These lighter gray bars represent our wild-type mice, and you can see their synapses have a high level of complexity, with the majority of the synapses have retinal terminals from 4 or more retinal ganglion cells. In contrast, these darker gray bars represent our mice lacking LRRTM1. And you can see these mice lose that complexity, with the majority of their synapses having only one retinal terminal. In addition, when we quantified the size of retinal terminals, we found interestingly that in mice lacking LRRTM1, in synapses of both types, they just tended to have larger retinal terminals. So once we had studied the role of these two proteins in retinogeniculate synapse development, we now had a really nice model to study these complex synapses. We had our mice lacking LRRTM1, which we knew lost those complex synapses, and we also had our mice lacking NRN1, which we knew had those intact synapses. So that led us to our next question: what role do these complex retinogeniculate synapses and this retinal convergence in the visual system play in visual behavior? So we decided to test visual behavior using a water maze. The water maze is constructed as you see here in this bottom diagram. Mice begin this task when they're placed in the bottom arm of the water maze, and they're trained to swim up towards these two arms. Each arm has a screen, and it displays either our positive visual cue, S+, which is that black and white grading, or it displays our negative visual cue, S-, which is a homogeneous gray screen. And mice are trained to associate the positive visual cue with the platform, which is hidden just beneath the water and allows mice to escape the water maze. We then alternate the location of the platform and the cue from right to left in these pseudo-random patterns you see up here. This allows us to test their visual behavior by looking at how accurately they are able to locate that platform. As you can see in this example, if the mice is able to locate the platform correctly, they'll swim directly towards our positive visual cue here. And because we're testing 3 groups, we have our wild-type mice, our mice lacking LRRTM1, and our mice lacking NRN1. We did our statistical analysis with a one-way ANOVA and Bonferroni post-hoc analysis. And in addition, we also added a 70% accuracy threshold to indicate behavioral significance. In other words, when mice drop below that 70% accuracy, we consider them to be failing the task. And that allows us to compare when mice are either dropping below that 70% accuracy threshold or rising above, and compare that between all 3 groups as well. So we first tested the ability of mice to actually learn this task. So as I said before, our positive visual cue is this black and white grading. Our training took place over 8 days with 10 trials in the water maze on each day. And as you can see, all 3 groups of our mice rise above our 70% threshold on day 3, and by day 8, they all have very similar levels of accuracy. As a negative control, we added Math 5 mice, which you can see represented by the squares in the bottom, And these mice are genetically blind. And for us to see that they're not able to learn this task confirms that there's no non-visual cues that could help the mice find the platform. We then tested the visual acuity of our mice. And we did this by altering our positive visual cue by increasing the frequency of those black and white bars, which makes it harder to distinguish between the bars and the homogeneous gray screen. Their baseline frequency is 0.17 cycles per degree, and we went all the way up to 0.64 cycles per degree. And as you can see from the lines, all three groups behaved very similarly, and they all dropped below the 70% threshold at 0.57 cycles per degree. And although you do see a statistically significant difference at 0.62 cycles per degree, because all three of those groups are below that 70% threshold, we consider them all to be failing the task, and therefore it's not a behaviorally significant difference. We then tested the ability of mice to detect different levels of contrast. So now, altering our positive visual cue, instead of changing the frequency of those bars, we changed the contrast between the black and the white bars. So their baseline is, of course, 100% contrast between the black and the white, and then we decreased it all the way down to 10% contrast. And now you can see all three groups perform very similarly again, all falling below our 70% threshold at 10% contrast. And again, we do see a statistically significant difference at 10% contrast, but again, all three groups are below that 70% threshold, so we consider them all to be failing at that level. We then asked, can the mice distinguish between horizontal grading and vertical grading to test their ability to discriminate two patterns? So for this, we replaced our negative visual cue with horizontal grading. And this is done similarly to our training modality. We trained this over 8 days with 10 trials on each day. And the question we were asking is, can they learn to identify the vertical bars instead of the horizontal bars? And again, all three groups performed very similarly. They all rose above our 70% threshold by day 4. And by day 8, they had very similar levels of accuracy. So far, all three of our groups have performed these tests very well, and we did not find any deficits in one particular group. So the next thing we decided to ask was, what is the sensitivity of these mice to detect pattern? So to do this, we actually used the same visual cues as our last test, the horizontal and the vertical bars, but now we increased the frequency of those bars as we did for acuity testing. We started with their baseline frequency of 0.17 cycles per degree, and we increased to 0.32 and 0.55 cycles per degree. And you'll notice in this graph, we also added increased trials at their baseline frequency to ensure that the mice are not just de-learning the task. And as you see here, when we increased to 0.32 cycles per degree, our mice lacking LRRTM1, which are represented by this darkest bar, they drop below our 70% threshold. And the other two groups of mice are actually still able to perform that task accurately. That's a statistically significant difference there. And when we increase again to 0.55 cycles per degree, now all of the mice are failing at this task. And this indicated to us that our mice lacking LRRTM1 actually had a decreased sensitivity for detecting pattern. So we tested this again in a slightly different way. This time, instead of altering the frequency of our two visual cues, we altered the contrast as we did in contrast testing. We started again with their baseline contrast of 100% and decreased to 50%, 25%, and 10% contrast. And you can see when we went down to 25% contrast, our mice lacking LRTM1, again represented by that darkest bar, they drop below our 70% threshold, whereas our other two groups are still performing fairly accurately. And then when we drop again to 10%, we see all three groups are now below that 70% threshold, although the mice lacking LRTM1 still are performing significantly worse. And this really confirmed our conclusion that mice lacking LRTM1 had that decreased sensitivity to detect pattern. So it's really been until recently that it was thought that information in the visual system traveled in those parallel pathways. And it's really this new idea that we found that there's that high level of convergence in the visual system introduced by these really complex retinogeniculate synapses where those multiple retinal ganglion cells are converging onto a single dendrite. So we investigated the roles of 2 proteins. We looked at NRN1, which did not appear to play a role in the development of those synapses, but we found that LRTM1 appears to be necessary for those complex synapses to form. We then took advantage of those mice to investigate the functional consequences of retinal convergence in the visual system, and found that mice lacking LRTM1 appeared to have a decreased sensitivity for pattern discrimination. And this is really the first link between convergence in the visual system and a behavioral outcome in visual behavior. So not only does this give us insight into the function and structure of the visual system, but this will also have potential implications for future regenerative therapies. I'd like to thank everybody in the Fox Lab. I wouldn't be here, standing here today talking to all of you without all the support and help I've gotten from all of these people here, as well as Dr. LeConte, Dr. Vaughn, and Lauren Harris. Thank you.
Time for a question or two for Gail. Dr. Freeland.
So I'm going to ask you a tough philosophical
Okay.
Why do you think nature goes to such great pains to establish in the retina all these different organizational structures of receptive fields, to have these parallel channels, and then to mix it up at the first synapse of the LP?
I mean, it's hard to know because I think we could even take that a step further and say we have different classes of retinal ganglion cells that we thought had different functional purposes, but then in these complex synapses, we also see multiple classes of retinal ganglion cells converging onto a single thalamic cell, whereas historically we would have thought those should all remain separate. So it's really hard to know, and I think it's one of those things that's just so subtle. And the outcomes we saw in our visual behavior were also subtle. It wasn't as if the mice were blind or couldn't do any of our tasks. So I think it's hard to know what that means at this point.
One other question, Natalia.
Hi, I was just wondering how the different levels of contrast that you were manipulating. I was just wondering how significantly reliable they are to test how the mice would actually see things in day-to-day life.
I mean, testing visual behavior in mice is in general difficult because obviously we can't ask them what they're seeing. So it's just one of those things where it's hard to know. Like, we devised all these different ways of testing, like not just acuity, but contrast sensitivity. I think the contrast sensitivity should be fairly accurate because we do use the homogeneous gray screen as our negative Q. And when we got down to 10% contrast, it was a fairly subtle difference, even just to my eye. So I think the fact that the mice did as well as they did with that task is pretty reliable.
Okay, let's thank Gail again. So our third presenter today is going to be Ankita Sridhar. She worked with Dr. Jack Perkins in the Department of Emergency Medicine, and she worked on a project that was evaluating whether cumulative organ dysfunction is a predictor of mortality in severe sepsis patients.
Yes, hello. Hi everyone, my name is Ankita Sridhar, as you just heard, and this is the title of my project. I worked with Dr. Jack Perkins in the ED. So to begin with, let's talk a little bit about sepsis. Sepsis is typically thought of as your body's response to a significant infection. It's typically thought of in this spectrum going from sepsis to severe sepsis to septic shock. And for the purpose of this research project and essentially this presentation, we're mostly going to be focusing on severe sepsis, which is defined as sepsis plus the presence of organ dysfunction. Sepsis is a very big deal in our healthcare system. It accounts for more than 1 million cases to the hospital and over 500,000 visits to the ED annually. And if you look at those mortality rates there, mortality for sepsis can be anywhere from 20 to 50%, anywhere in that sepsis spectrum. It's such a big deal that the government got involved. And by the government, it's the CMS, or the Center for Medicare and Medicaid Services. In 2015, they issued what was the SEP1 Core Management Bundle, looking at severe sepsis and septic shock patients. The purpose of this bundle was to provide a standard of care for patients with severe sepsis and septic shock the minute they hit the ED. And this bundle had two prongs to it. There was a diagnostic component, but also a management component. And this was to provide this standard that all hospitals could abide by the minute anyone walked into the ED with severe sepsis or septic shock. It's important to note that this bundle was made from the recommendations of the Surviving Sepsis campaign of 2012. So let's take a step back and look at organ dysfunction, because organ dysfunction is a key part of defining someone as severe sepsis. Organ dysfunction has been looked at for generations, especially in the context of sepsis, and especially when you're looking at assessments like the SOFA score, the sequential organ failure assessment, or the APACHE or the MOD score. But it's important to note that these scores, particularly the SOFA score, they're validated in the ICU setting or the inpatient setting, not so much in the emergency department setting, because you need a lot of variables when you're looking at these, when you're looking to calculate these scores. It's not so great when you're trying to triage someone when they walk into the ED. Furthermore, the SEP1 management bundle, they used 6 different organ dysfunction qualifiers, not coming from the SOFA score, but coming from the Surviving Sepsis campaign of 2012. These organ dysfunction qualifiers, in fact, are binary, so it's really a yes or no question. Does this patient meet this qualifier or does it not? It's really not looking at a grade of 0 to 4. So when we look at severe sepsis as defined by SEP1, you have documentation of a suspected infection, you have an elevated temperature, heart rate, respiratory rate, and white blood cell count criteria indicating the presence of what we call SIRS, or systemic inflammatory response syndrome. And then you have the presence of organ dysfunction that's qualified by the evidence of any one of 6 organ dysfunction qualifiers. If we take a look further at our organ dysfunction qualifiers, you have 6 different criteria representing kind of an overall look at your body system. So you have systolic blood pressure, looking at your cardiovascular system. You have an elevated creatinine, looking at your kidneys. You have bilirubin, looking at your liver. You have thrombocytopenia and INR, looking at your hematology and your coagulation system. And then you have elevated lactate greater than 2, and that's just looking at overall tissue hypoperfusion. So these qualifiers combine a multi-system approach, but again, they're binary. You either have it or you don't. So why is this important? Well, these core measures put out 6 different organ dysfunction qualifiers, and the presence of any one of them means you have severe sepsis. This is almost a one-size-fits-all approach for patients. That means that when Miranda walks into the ED with just one organ dysfunction qualifier, and when Mark walks in with all 6 organ dysfunction qualifiers, well, hey, both of them have severe sepsis. And that means that there are mandatory treatment benchmarks for both of these patients, Miranda and Mark likely. Which begs us to ask, well, is the mortality risk the same for all of these severe sepsis patients? And is there an efficient way that we can risk stratify the minute they walk into the ED on their initial presentation? So our research project is 2-pronged. First, we ask, well, how does cumulative organ dysfunction predict mortality on initial presentation to the ED? And then we look at, well, how does this compare to the individual organ dysfunction qualifiers? Remember, there were 6 of them that the CMS put out. And with either of these, well, can we create a form of risk stratification that we can use on initial presentation to the ED? And we hypothesized that yes, it's going to be cumulative organ dysfunction that will be the direct predictor of mortality, and we should be able to use this cumulative organ dysfunction to create a risk stratification method on initial presentation to the ED. So this diagram kind of represents our data flow. We began looking at 519 patients who triggered a sepsis alert in the CRMH ED. And just a quick refresher on sepsis alert, it was a program that was designed in our CRMH ED to identify patients who are at risk for sepsis immediately on presentation to the ED. And this could be patients who have sepsis plus hypotension, and elevated lactate, but also just clinical provider suspicion for sepsis. And looking at these 519 patients, we used a strict exclusion criteria to make sure that these patients had sepsis on admission, did not have septic shock, and then actually did have any form of organ dysfunction qualifier based off of these CMS core measures. And we ended up with 289 severe sepsis patients that we included in our final data analysis. We had a lot of data variables when we were looking at this project. We had data variables that were both automatically extracted as well as manually extracted. Automatic extraction means data variables that are very easy to pull from a computer algorithm. So we're looking at variables like age, gender, vital signs. Those are things that a computer can pull out for you. And then we're looking at more manually extracted variables like, hey, did the patient need a pressor? And this could be anything from norepinephrine, this could be vasopressor, so there's a little bit more variability there. And then also 30-day mortality status. And so the manually extracted variables, those were gotten by 4 trained researchers who were part of the research team. And it's important to note this because ultimately inter-rater reliability was calculated amongst these 4 researchers, and it was calculated to be a moderate inter-rater reliability. So here we see our initial characteristics of our data set. As you see, the median age is about 65 years of age, which is as is expected, as elderly patients are at a higher risk to become septic. And if you look at our gender distribution, it is a pretty even distribution of male and female, as gender has not been shown to be a significant predictor of sepsis at all. So when we look at organ dysfunction, we find that, well, majority of our patients present with less than or equal to 3 cumulative organ dysfunction. That means that only 13 patients in our data set actually had 4, 5, or 6 levels of cumulative organ dysfunction. And then when we look at the individual qualifiers themselves, we find that elevated lactate greater than 2, was the most common organ dysfunction qualifier. That means that 76.8% of patients who came into the ED and were classified as severe sepsis had an elevated lactate. When we look at our primary analysis, and this is the logistical regression model that we ran for cumulative organ dysfunction and the individual organ dysfunction qualifiers, we found that cumulative organ dysfunction, in fact, was a direct predictor of mortality in our severe sepsis patients. And you can see that here represented in our forest graph right there. And we found that with every increase in cumulative organ dysfunction, there is a 1.57 times increase in their mortality rate. But what we also found was that no individual organ dysfunction qualifier was a significant predictor of mortality. Remember how 76.8% of patients with severe sepsis had an elevated lactate? Well, lactate is still not a significant predictor of mortality there. It's just cumulative organ dysfunction. When we take a step further and we look at the mortality levels at each level of cumulative organ dysfunction, we see that, hey, mortality does increase with increasing levels of cumulative organ dysfunction. Well, we already found that out with our logistic regression model. But what is important to note that we do have that low N for patients with 5 or 6 cumulative organ dysfunction. In fact, we only had 1 patient who had 5 or 6 levels of cumulative organ dysfunction. This makes it a little hard to draw robust conclusions about patients on this end of the spectrum. But what we could do was split it and look at it as patients with less than or equal to 3 and greater than 3. And when we did that, we found that patients with greater than 3 organ dysfunction qualifiers have a significantly higher mortality than their counterpart of less than or equal to 3, but also their remaining severe sepsis cohort. And in fact, their mortality is so high, it's similar to those of septic shock patients. And septic shock, in our research project, was defined by the use of pressors immediately in the ED. So these patients, when they have greater than 3 organ dysfunction qualifiers, their mortality rates significantly increases. I'm going to take a step back and address limitations, because there's always limitations in any research model. The big limitation for us is our study population. We are looking at one level 1 trauma center, our CRMH ED. However, it is important to note that our initial characteristics were similar to previous studies that looked at severe sepsis and sepsis patients. Another important thing to notice is the low N at 5 and 6 levels of cumulative organ dysfunction. We only had 13 patients that had greater than 3 cumulative organ dysfunction who presented to our ED in our time frame. But I would like to add that with our 13, we were able to reach statistical significance when comparing greater than 3 and less than or equal to 3. Another big thing is inter-rater reliability. Whenever you're using subjective variables and 4 different researchers, it's important to calculate inter-rater reliability amongst the researchers. When we calculated our Fleiss kappa analysis, we had a Fleiss kappa of 0.56, which indicated moderate inter-rater reliability. Not the worst, but also not the best. You could always aim for more inter-rater reliability. And this could be done in multiple different ways, whether that's further training your researchers, making a more strict algorithm for what defines a sepsis or not sepsis, but also choosing different criteria for what you're choosing reliability on. Now that we've addressed the limitations, let's summarize. What have we learned? Well, cumulative organ dysfunction is a direct predictor of mortality, and in fact, with increasing cumulative organ dysfunction, we have a 1.57 times increase. That type of organ dysfunction doesn't matter. It doesn't matter what qualifier they have, it just matters how many they have. And risk stratification is a possibility with these CMS core measures. Risk stratification is possible for patients with greater than 3 cumulative organ dysfunction. Let's go back to Miranda and Mark, why don't we? So Miranda, who came in with just 1 organ dysfunction qualifier, and Mark, who came in with all 6 organ dysfunction qualifiers. We've shown that, hey, the mortality risk is not the same between these 2 patients, and risk stratification should be explored for patients with greater than 3 organ dysfunction qualifiers, And risk stratification should be explored for Mark versus Miranda, because Mark's mortality rate is significantly higher than Miranda's from the minute they walk into the ED. So future directions for this project is largely looking at the risk stratification. There should be risk stratification that's explored using these particular core measures if CMS is enforcing hospitals to do that. And that can be anywhere from aggressive resource allocation within the ED itself and during their time course there, or even outside of the ED when you're looking at admission from the ED into the hospital. Furthermore, there's a lot of work that can be done looking at those individual qualifiers. We found that 76% of patients with severe sepsis have an elevated lactate level, but an elevated lactate level of greater than 2 is not a direct predictor of mortality in these patients. So there's a lot to be looked at with seeing, well, how do these individual organ dysfunction qualifiers even play if you're turning it into a binary yes or no qualifier? Last but not least, I'd like to acknowledge everyone in my research team, particularly Dr. Perkins, without whom none of this project would have been possible. I'd like to thank the remaining members of my research team, Dr. Henry Weigel, Dr. Mohsen Chowdhury, and Ben Bain. The 4 of us comprised of the 4-member research team who did all the data abstraction, and amongst the 4 of us was the inter-rater reliability. Dr. Yan Zhang, who is a part of Carillion Health Analytics, who helped pull the automatic data poll for all the automatically obtained variables. And of course, Dr. Allison Teggy, who helped me with the statistical portion of this and how to use R. And last but not least, of course, Dr. LeConte, Dr. Vaughn, and Lauren, without whom this research domain would not be possible. We'll take any questions.
Questions for Ankita? Lindsay.
Yes, so actually I went through all of the patients who did die in the emergency department. We had 3 patients who died in the emergency department, and this was done because all 3 patients actually had care withdrawn from them during the course of the emergency department. So it wouldn't have been a realistic situation to include them in the analysis and have possible risk stratification. As for in our subset, all 3 patients had family withdrawal care.
So you mentioned early in your presentation that one of the criticisms of the sepsis criteria is that they are binary. Do you think that reanalyzing the data that you have on the basis of the correlation between, say, a very high lactate level and not just this binary greater than less than 2 would increase the odds ratio for mortality observed with those system-specific markers?
Great question. I actually had the opportunity to look at that itself in a kind of looking at a different direction on this research project. When you do look at lactate as a linear variable, it regains its predictive ability. And in fact, I compared linear lactate, lactate greater than 2, and lactate greater than 4, and it regains its ability to predict when you go to lactate greater than 4 and when you have it as a linear variable. I was not able to perform any sensitivity or specificity analysis on that, but I did think it was interesting that creating that binary cutoff of lactate greater than 2 is where it loses its predictive ability. Yeah.
Do you think if you have a larger sample size that some of those individual variables, even at the cutoffs that were used by CMS, would gain mortality?
It's absolutely possible. I can't say anything what would happen if the sample size was a little larger, especially with the biggest example I can give is lactate, because lactate has been looked in so many different studies. Lactate greater than 2, if a patient comes in with lactate 2 as a 2.1 and that's their only organ dysfunction qualifier, what could that mean? And that could be sepsis, that could be so many different things. And I think it's just such a wide net that binary organ dysfunction qualifiers are casting that it's impossible to know when you'll achieve significance. I think the rationality behind kind of creating such a low lactate level specifically is to maximize specificity, at least when you're looking at sick patients in the ED, even though you're kind of losing out a little bit on sensitivity. But if you're using that as a diagnostic criteria rather than predictive for mortality, that's when you kind of have to use the bargain.
So our last presenter for this session will be Quan Fung. He worked with Dr. Warren Bickel and Dr. Sarah Snyder, and many of you are familiar with the concept of delay discounting, and he's going to talk about sort of a variation on that theme in terms of effort discounting. Go ahead, Quan.
Thanks. All right, hi everyone. My name is Quan, and I'll be presenting on this study right here. And I did this as part of the Addiction Recovery Research Center under the guidance of my mentors, Dr. Warren Bickel and Dr. Sarah Snyder. And overall, our large theme of the lab is trying to understand the mentality of individuals who have addictions, and a big part of that is understanding decision-making and how people make choices. So with that, I want to offer everybody a choice between receiving $50 right now versus $100 in a year. And let's say you chose the $100 in a year. I can increase that offer again to $75 now, and let's say you still chose the $100 in a year. So if I increase the offer again to $87, let's say you chose the $87. So what that tells me is somewhere between that $75 and $87, you are indifferent between receiving a smaller amount of money now versus a larger amount of money later in the future. And so that idea of being willing to take something less sooner is probably not too unusual for you. And what that refers to is a concept called delay discounting, and that's the tendency to devalue rewards following a delay. So if I kept asking you those sets of questions across different time points, I can calculate your indifferent points. From there, I would be able to characterize your delay discounting behavior. I would also be able to model this and get different variables, including this K value, which would describe your discounting rate, which would be your unique discounting preference. So some background about this. So delay discounting is seen across individuals. So you see this with healthy individuals as well. However, delay discounting is more prominent in individuals who have addictions. So even though this graph is specific to opioid-dependent individuals, this is seen across addictions, including morbid obesity, gambling, and alcoholism. And then, in addition, this suggests that addicted individuals may have a sort of altered, shortened sense of time where they're willing to take short-term benefits at the expense of long-term consequences. So, our overarching research question is, how can we better understand behavioral decision-making processes that may help patients recover from addictions? And so this study looks at both delay discounting and effort discounting, and there are different schools of thought related to effort. One prominent idea is effort discounting, which is the tendency to devalue rewards following greater effort to obtain that reward. So if you put in more effort to get something, then you may have less subjective value for that reward. So for example, if I were to offer you some money for doing no work versus more money for doing a little bit of work, if you had higher effort discounting, you'd be less likely to choose the option that requires more effort. So some studies have suggested that there is some type of relationship between delay and effort discounting, and most likely these studies suggest that these are overlapping but separate and distinct processes. And although there has been a good amount of research related to delay discounting and addictions, there really hasn't been much done related to effort discounting and addictions. So our research question for this study was, how does effort discounting compare to delay discounting behavior among individuals who drink alcohol? And then how does this behavior change with different rewards, specifically money and alcohol? And we believe that if you had higher alcohol use disorder severity, you would have both higher delay and higher effort discounting. So this is the methodology of the study. We surveyed 100 participants on Qualtrics, and then we fielded the survey on Amazon Mechanical Turk. The MTurk website is essentially an online marketplace where people can be paid to do different tasks, including filling out different surveys. So to be eligible for the study, you had to be at least 18 years of age, be located in the US, drink some alcohol, and then have a certain approval rating within the MTurk site. We also asked participants to fill out questions regarding their demographics, alcohol use, and computer use. And alcohol use was measured in both DSM-5 criteria as well as audit criteria. So the DSM-5 uses 11 questions, and the more positive responses you have, the greater your alcohol use disorder would be. And then we had participants complete in randomized order 4 discounting tasks that involved either delay or effort, and in which the reward was either money or alcohol. So just to make this a little bit more concrete, I included the first question from each of these tasks. So with our delay discounting task for money, we asked, would you rather have $50 now or $100 in 3 weeks? And then we included a version of this task where the reward was alcohol. So would you rather have 10 drinks now or 20 drinks in 3 weeks? And I'll get to a little bit later how we got to those numbers. And then in addition to these delay tasks, we also had a version of these tasks involving effort. So would you rather have $50 for typing 0 words or $100 for typing 504 words? And then lastly, we had a version of this task with alcohol as a reward. So would you rather have 10 drinks for typing 0 words or 20 drinks for typing 504 words? So in essence, these tasks are very similar with one major difference in between each of them. Okay, so I'm going to go into a little bit more detail about these tasks. So with our delay discounting tasks, we asked participants a series of questions pretty similar to the questions I asked at the beginning of the presentation. And from the responses, we were able to calculate this K value, which describes the rate of change of this discounting curve. And then in addition to that, we created our own effort discounting task. So instead of asking participants to wait for a reward, we asked them how many words they would be willing to type for a reward. And we took the delay and the delay discounting task, and we converted that on a one-to-one basis to the amount of words that we asked participants to type. So for example, a 1 hour delay would be equal to 1 word, and a 1 week delay would be equal to 168 words. So this was done so that the scale of these tasks would be proportional to one another. And then in addition, we also asked participants how many alcoholic beverages they considered to be equivalent to $100. And then we made sure to use their responses so that the amount of alcohol offered in the alcohol reward test would be equivalent to the amount of money offered in the monetary reward test. So all of this is to say that all 4 tasks were designed so that they would be comparable to one another. So here are our results. We see in these 4 panels our 4 discounting tasks. And then on the x-axis is the DSM-5 criteria. So higher DSM-5 would mean greater alcohol use disorder severity. And then on the y-axis is discounting. So that would mean higher delay or higher effort discounting, which would mean less willingness to wait or to work respectively. And we see that there is a positive correlation between DSM-5 and delay discounting for money, which means that if you have more alcohol in use, you're less willing to wait to get money. We also see this positive correlation between DSM-5 and delay discounting for alcohol. So if you had more alcohol in use, you were also less willing to wait to get alcohol. We didn't see any correlation between DSM-5 criteria and effort discounting for money. So all these individuals were similarly willing to work for money. But there was a negative correlation between DSM-5 and effort discounting for alcohol, which means that they were more willing to work for alcohol. So just taking a step back, what this means is that participants with greater alcohol use disorder severity had higher delay discounting for both money and alcohol. So across different types of rewards, these participants are more impulsive, and they're more impulsive, and that's pretty consistent with what we see in the literature. And then alcohol use disorder severity was not significantly associated with effort discounting for money, but it was more associated with effort discounting for alcohol. So this shows us that there's a differential after discounting preference for different types of rewards with an increased preference for your drug of choice. So we were wondering why we were actually seeing these effects, and we were trying to figure out, is there a mechanism that can explain what we're seeing? So we believe that these effects could be driven by the participants' demand for alcohol. So we thought that individuals with greater alcohol use would have greater demand for alcohol, and therefore would have an increased willingness to work for alcohol. So we wanted to test this, and so we actually conducted a follow-up experiment. And then this follow-up experiment replicated the methodology for experiment 1 with the addition of an alcohol purchase task. So in this task, we asked participants to imagine being in a scenario, and we asked them how many alcoholic drinks they would purchase at different prices. And from that, we were able to assess their demand for alcohol. We used a total of 411 participants as part of this analysis after checking for financially consistent responses. And then here are our results. So in essence, we were able to effectively replicate the findings that we saw in experiment 1. So increased use of alcohol was associated with less willingness to wait for either money or alcohol. There's no association with willingness to work for alcohol, but an increased association with the willingness to work for alcohol. No association to work for money, but an increased association to work for alcohol. So this was a positive finding in the sense that we were able to essentially replicate our findings, but we did this with a larger sample size, so that was very reassuring. So what the study that added that was new is that now we have all these participants and we have information about their demand that we can therefore correlate and correspond to their discounting preferences. So we measured this with our demand parameters Q0 and alpha, and Q0 is a measure for initial demand, so how much you would want of a commodity if the price was 0. This has also been referred to as amplitude or intensity. Sometimes I think about it in terms of like craving or strength of your craving for something. And then alpha is a measure for demand elasticity. So it's also known as price sensitivity. So if you're very price sensitive, as the price for something increases, you're going to want that less. You're going to want that less quickly. And that has also been referred to as persistence. So now we have, what this gives us is that we have two variables that gives us a specific way to look at demand in a quantifiable way. So what we found were that, yes, these two demand parameters were significantly correlated with our variables of interest. Greater initial demand for alcohol was associated with greater willingness to work, greater alcohol use, as well as a greater willingness to work for alcohol. Price sensitivity was negatively correlated to alcohol use disorder and positively correlated to effort discounting. So that means if you were very price sensitive, you tended to drink less and you were less willing to work for alcohol. So I know there are some double negatives in those statements, but overall what that tells us is that greater demand for alcohol was associated with greater alcohol use and a greater willingness to work for alcohol. So this shows us that there is a correlation between all these variables, but we wanted to take this one step further and we wanted to see if demand was actually driving what we were seeing. So we conducted 2 separate mediation analyses, and then just taking a step back, if alcohol use disorder was directly related to effort discounting for alcohol, you would see this very linear direct pathway between them. However, you can also have a mediating variable that in essence takes this effect right here and helps transform it in a way that produces our outcome. So when we tested that with Q0 and initial demand, we saw that it did have a significant mediating effect, and it actually accounted for 37.8 of the total effect that we were seeing. Alpha, on the other hand, the price sensitivity, even though that did have some explanatory power, it was not statistically significant. So what that tells us is that we saw this initial relationship between alcohol use and willingness to work for alcohol, and now we can say that demand did have a mediating effect and was helping to drive some of what we were seeing. Okay. So in summary, what this study suggests is that both delay and effort discounting are processes that contribute to negative decision making. So increased use for alcohol was associated with less willingness to wait for either money or alcohol, but it was not associated with the willingness to work for money, but it was associated with the willingness to work to get alcohol. And these results are explained in part by increased alcohol evaluation, particularly with increased initial demand for alcohol. So together, what these results say is that these individuals were actually in a really risky, it paints a really risky picture where these individuals are both more impulsive but also more willing to work to get their drug of choice. Just some limitations to consider. So the DSM-5 criteria are really more recommended to be conducted in an in-person setting with a trained professional, but that wasn't really feasible with the MTurk site. And then participants may have also had some difficulties in interpreting a theoretical task and a theoretical reward. So in the future, we want to consider extending this work to non-hypothetical tasks and rewards. And then we also want to consider including people who don't use alcohol as well as individuals who abuse other substances. So a major goal of our study is trying to figure out what are the meaningful interventions that can create enough of a barrier that can change behavior related to delay or effort. And so the BICL lab does not actually endorse a product like this, but we believe that potentially using things like specialized containers, alternative reward cues, asking participants to engage in physical or cognitive tasks before receiving a reward, or using time locks that would incorporate both an element of delay and effort could be helpful. And ideally, we believe that understanding both delay and effort discounting could help us develop interventions that can utilize both of these factors as a way to help with addiction recovery. With that, I just want to thank the co-authors of the study, Dr. Bickle, Dr. Snyder, and Dr. Teggy. This study was actually just published a few weeks ago, so I just want to say a big thanks to all of them for really helping push that through. And then a big thanks to the participants in the study, and then a thanks to everyone at the Addiction Recovery Research Center for allowing me to research with them over these past 4 years. And just like to open up for questions or feedback. Thank you.
Questions for Quan?
Oh, Cliff?
How demographically comparable were the groups that had alcohol, severe alcohol use disorder versus those with relatively few criteria met?
So originally we had a different design for the study. So we had kind of like low alcohol use was higher alcohol use, and those two populations were pretty similar demographically. But we changed the study so that instead of having two groups, we used a continuum from DSM-5, 0 to 13. And so at that point, it didn't really make sense to kind of like look at the demographics across these groups necessarily, because it's more of a continuous variable rather than two different groups that we're comparing. But originally they were comparable.
How did you entice people to participate in the study?
We offered them the reward of $2 to do the study, which was actually a pretty good rate compared to other surveys on the MTurk site, considering that it was a relatively short study as well.
Do you feel that having a financial incentive to participate in a short study might artificially affect your results of looking at people who are motivated by financial reward?
Yes, that's a very good consideration. So I guess in the future we can look for people who do not want a financial reward to complete our study as well. So thank you. All right, thank you.
Okay, before I let you all run to the next poster session, I want to remind you that there is a survey out there. Dr. Mutchison says very few of you have participated, so we don't have a lot of votes for the student awards for the posters. So go out there and vote for things like most tweetable poster, something about most persistent research projects, so required like the most amount of work to do. So some fun categories, make sure you get those in before the beginning of the next oral presentation, please. And then we'll announce those awards at the end of the oral presentations. Thank you.
phenotype were more willing, maybe they are more likely to actually already be at risk of addiction or even already. But you probably couldn't tell that from looking at this, right?
Yeah, so my history is, so I think
registration. I need to go down and get the certificates. So they're here. Yes, you just have to open them. Andrews is not. It's just here.
And then to start it in interview or whatever, what do I do?
So
It's just like normal.
yeah, it's just like normal. It just looks different, but yeah, it's just different. They did the expanded screen. Yeah, just escape out of it. They'll take you there. But I'll be here for the first two. Walk out for the next two, and then I'll be back so we can do a quick sign. Yes, a quick sign. And Danielle is helping me with this part, and I've already done the sign up sheet, and so they'll be out at the end. Yes. We should be back. We have more votes this year than last year. Oh, is that very impressed? Yeah. And maybe before you start, you can maybe complete the voting, because I think it's like 24 were pending. So then you need to go ahead and complete it. Yeah. No. But they kind of know something. They're like, where the sheep? I said, why?
Okay, everyone. I think we're going to go ahead and get started. It's a little bit after 3:30. If I can have everybody take their seats. Remember not to be afraid of the front rows. I was up here all by myself for the first half, so I don't bite. Yeah. All right. So I have the pleasure of being your hostess through the latter half of the day for 4 additional great presentations. And without further ado, I'm going to announce our first presenter. This is Miss, where is my list? I know your name, by the way. This is Lindsay. I just want to make sure I'm keeping up with her title. This is Miss Lindsay McGuire, who worked with Dr. Robin Queen at the Granada Biomechanics Lab at Virginia Tech. Lindsay's talk is entitled The Impact of Activity Level on Loading Symmetry During Return to Sport Hop Testing in Healthy Participants. So I'm going to let her take it away.
All right, can everyone hear me? Great. So, talking a little bit more broadly in this, I'm just going to be talking about evaluating loading symmetry in general during hop testing in the clinical setting. Like Dr. Vaughn said, I was working with Dr. Robin Queen for the last 4 years in the Granada Biomechanics Lab at Virginia Tech. So a little bit of background here. Loading symmetry is one measure that's used to evaluate readiness to return to sport, to go back to playing a sport after injury to an anterior cruciate ligament, like this one Tom is undergoing. ACL injuries are quite common, and up to 300,000 ACL reconstructions are performed every year in the United States. Second injury rates after an ACL reconstruction, depending on the study you look at, range from between 9.6 to 25%. We believe that a large part of the second injury rate is due to the fact that injury and reconstruction cause asymmetry in movement and loading patterns. And this loading asymmetry means the risk is increased on both the surgical limb and the non-surgical limb. That high re-injury rate tells us that there are a lot of athletes who aren't properly rehabilitated by the time they're told they're safe to go back to playing by their physician. What is problematic is there are no universal guidelines that inform return to sport decision making, and different physicians use different benchmarks in evaluating their post-operative patients. So return to sport hop testing is one tool that clinicians can use to identify asymmetry in the clinical setting. In particular, the single hop for distance is the most commonly used hop test in return to sport evaluations. Hop testing involves 4 hopping tasks: the single hop, the 6-meter timed hop, the triple hop for distance, and the crossover hop for distance. Hop testing, unfortunately, only gives us information on symmetry in distance hopped, which has not proven sensitive or specific in identifying who is safe to play and who is at risk for a second ACL injury. So we think that objective information regarding movement quality is necessary. So can we test movement quality in the clinic? Traditionally, testing movement quality involves having special equipment like force plates, which are installed in the ground, they're expensive, or motion capture, which is technologically difficult to learn and also time-intensive and expensive. The load sole, pictured here, is a single sensor insole that can be slipped into a patient's shoe and provides real-time data on the force between the foot and the shoe. So the purpose of this study was fourfold. First, to validate the load sole for use in hop testing in the clinical setting. We hypothesized that the load sole would be valid when compared to force plates and would be repeatable between days. Next, to explore normative values for loading symmetry in a healthy population, determine what exactly is normal for our measures. To identify whether there are any sex-specific or activity-level-specific differences in a healthy population, we hypothesized that there wouldn't be any differences that were attributable to either sex or activity level. And finally, to provide a pilot analysis of how ACL reconstructed patients differ from healthy controls in loading symmetry. And we hypothesized that the ACL reconstructed patients would produce less force on their surgical limb, resulting in a lower limb symmetry index, which we'll talk about later, compared to the healthy controls. So the first piece of this was that validity and repeatability piece. We recruited 30 athletes who were recreationally active for at least 30 minutes, 3 times per week. They were fitted with the load sole insoles and asked to perform 7 single hops for distance on each leg onto force plates, as shown here. So they're wearing the load soles and hopping onto the plates so that peak force measurements during the landing phase of the hop could be measured simultaneously with both instruments. 15 of those participants returned for a second day to test the repeatability of the load sole. And then peak force measurements both between instruments and between days were tested for consistency using two-way mixed interclass correlation coefficient. Then we looked at the bias for the load sole for either underestimating or overestimating peak force and visualized that using Bland-Altman plots, which fortunately Omar has already showed us today. So first we compared the measurements, like I said, from the load sole against the measurements that were simultaneously recorded from the force plate. And we found a between measures ICC, or interclass correlation coefficient, of 0.707, which indicates good correlation. Next, we compared the measurements from the load sole taken on each of 2 separate testing days, and we found an ICC of 0.876, which indicates excellent correlation between days. From these measures, we gathered that the load sole is both valid when compared to force plates and repeatable on 2 separate days. Next, to visualize bias, like I said, we used a Bland-Altman plot. For the validity measure, we had a negative bias in the load sole of 0.46, which is visualized here in this thick black line. And then the gray lines show us the 95% confidence intervals for that measure. There was no bias that we saw, no significant bias in the between-day measure. So next we looked at characterized loading symmetry in a healthy person. We recruited 52 people here who are healthy, recreationally active, again, at least 30 minutes, 3 times per week. And here we show our participant demographics. We had an even split between men and women, 26-26. Males and females did differ based on height and body mass, as one might expect. But there was no difference in average age or in average activity level as characterized by a MARC score, which is a self-reported scale with a range of 1 to 16 measuring activity frequency. So we defined our high activity group in this as a marks of 9 or greater, so 9 to 16, which is where we would expect the majority of ACL reconstructed patients to fall. So in addition to 7 single hops for distance, our participants performed 3 repetitions each of a triple hop, which looks like this, and 3 repetitions of a crossover hop, which looks like that. So they did that bilaterally on each leg while wearing the load sole insoles. Then we calculated an average limb symmetry index for each task, wherein the peak force produced by the non-dominant limb was divided by the peak force produced by the dominant limb, multiplied by 100%. So this means a high limb symmetry index indicates that more force was produced by the non-dominant limb and vice versa. For our ACL reconstructed patients, limb symmetry was calculated a little bit differently. It was peak force produced by the surgical limb over peak force produced by the non-surgical limb times 100%. For our healthy controls, limb symmetry indices were compared between men and women and compared between participants of the high activity level group versus the low activity group using independent samples t-tests. So what we found is on average, which is all of these, our participants were roughly symmetric. So all of our limb symmetries for all of these groups were hovering right around 100. The orange is showing the entire group. Maroon is our male population, and then yellow is the female population. And there was no significant difference in limb symmetry when comparing males and females. Then looking at our activity level group, again, there were no significant differences between groups, and everybody was right near that 100% on average. So how symmetric are we? We just saw on the last slide that our average participants looked like they were about 100% symmetry. But there was a large amount of variability in what could be considered normal limb symmetry. So for example, only 58% of our healthy participants had a limb symmetry index of 90 or better during a single hop. And then only 35% of participants had a limb symmetry index of 90% or better during a crossover hop. So what's important here is that 90% symmetry is often used when testing hop distance as a benchmark to figure out who's ready to go back. If you can reach 90%, you're healthy. So 90% when using these measures may not be a realistic goal for an athlete trying to rehabilitate. It isn't until a limb symmetry index of 60 that we see all or nearly all of our participants reaching the same limb symmetry goal. So here we provide a pilot analysis of peak force loading symmetry measured by the load sole in an ACL reconstructed population as a proof of concept. So we recruited 5 patients who had undergone an ACL reconstruction, who had been returned to sport. They came in and completed the 7 single hops, 3 triple hops, and 3 crossover hops while wearing the load sole insoles. And what we found, unfortunately, was loading symmetry in this group was quite varied, and we weren't powered to find trends among these patients. A study published from our laboratory last year in Medicine and Science in Sports and Exercise did examine kinetic measures during hop testing in a larger ACL group. So here I've pulled that information for those of you dying to figure out what the answer is to that question. So this looked at 25 ACL reconstructed patients. You can see the average demographics here, and the MARC score, like we thought, is significantly higher than in our healthy control population on average. So when compared to matched healthy individuals, our ACL reconstructed patients didn't differ significantly in peak impact force for a single hop, which is this one, but did in a triple hop and a crossover hop. They did land with less impact force on their injured limb during those 2 hopping tasks, which are the more technically difficult tasks than just a single hop for distance. So what have we learned? The load sole is valid and repeatable for measuring limb symmetry in a clinical setting. There were no differences that were attributable to either sex or activity level, meaning that these factors don't need to be considered in return to sport evaluations. Peak force loading symmetry in healthy athletes has high variability. Baseline symmetry level might be a better rehabilitation goal than a generalized goal, especially that of 90%, which means that if we can test symmetry in people who play sports that put them at a particular risk for ACL injury before they have a chance to get hurt, that personal normal symmetry level is likely a much better goal than a generalized normal goal. And finally, we believe that measuring symmetry in movement quality clinically will better inform our return to sport decision making. Future directions, we want to identify which task or which battery of tasks is ideal in identifying patients at risk for a second ACL injury so that we can determine the simplest way for our clinicians to identify patients who are safe to return to sport versus who are not safe to return to sport and are at risk for a second injury in an evidence-based manner. I have a few people to thank for this. First of all is Dr. Queen, who has put up with me for 4 years. The other members of the Granada Lab, specifically Alex, Kristen, and my classmate Adam, Dr. LeConte, Dr. Vaughn, and Lauren Harris, and Lauren Harris for guiding us through all 4 years of research domain, Ryan Anderson for his help with the videos, and each of my peers who participated in the study. Thank you all. At this point, I'll take any questions.
Okay, we have time for a couple questions.
I answered everybody's questions. Yes?
So how did you assess for effort between participants to make sure that maybe they weren't just trying to guard against the surgical versus if it was actually preventive?
So that's a great question, because one thing that ACL reconstructed for people who want to go play their sport do, especially in hop distance studies, once they learn what the goal of the task is they will hop less far on their good limb, so that when they try on their surgical limb, they'll get a good symmetry. So the most important thing that we do is we don't tell them why. We don't tell our participants why we're asking them to do what we're asking them to do. The other part of that is I was the one doing this, and I kind of cracked the whip on most of our people so that they would actually try. Ashley knows. Anyone else? Okay.
So our next speaker is going to be Ms. Lambi Lee, who has been focusing her research on a topic near and dear to my heart. She's been looking for targets to develop new therapies in glioblastoma. And I'm sorry, she's been working with Dr. Ji Sheng. Really quickly, the one thing that I was asked to do, which I totally failed at when I got up here before, I'm supposed to ask you to complete your voting for the posters outside. So if you haven't done it, hit enter, because I think the time's up. So let's get those in.
Oh, I forgot to do that. Hi, everyone. My name is Lambi. Today I'm going to be talking to you about this gene called PIK3CB, and specifically how it may allow us to explore precision medicine in glioblastoma. So, as we all know, glioblastoma is a very deadly disease with a poor prognosis. Treatment options are extremely limited and include surgery, radiation therapy, and chemotherapy with temozolomide. But despite these aggressive treatments, still 90% of patients experience recurrence within 2 years. And after that, a survival is expected to be only about 5 to 7 months. So because of this, we really need new treatment options for glioblastoma. So what about precision medicine? Well, precision medicine says that patients with tumors that share similar genetic changes should receive treatment specifically for that type of change. And we already actually have this in treatment of cancer so far. So imatinib for CML and GI stromal tumors. We have Herceptin for HER2-positive breast cancer, as well as erlotinib and gefitinib for lung cancer. So why not glioblastoma? Well, we just don't have that information yet until now. So in order to get you to understand what gene I'm focusing on, PIK3CB is involved in this PI3K pathway. So imagine that you are on the cell membrane and you have a receptor tyrosine kinase that responds to an external growth factor. This results in recruitment of two subunits called P110 and P85. Now this complex here is representative of an activated PI3K kinase. So this activated kinase then goes on to activate downstream signaling factors such as AKT, which then promotes cell survival and inhibits apoptosis. So as you can imagine, dysregulation of this pathway can result in unregulated cell growth, and in fact is what you find implicated in many different types of cancers, including glioblastoma. And actually, PI3K inhibitors have been tried in many different studies before and in clinical trials. In fact, we've used pan-PI3K inhibitors, which inhibit this entire kinase here. And this was shown to have poor tolerance in clinical trials just because there's so many adverse side effects associated with it. But what we're beginning to learn now is that there are many different isoforms that exist. Specifically, within this P110 subunit, there are 4 different isoforms: an alpha, a beta, a delta, and a gamma isoform. And all of these are present in cells in different quantities. So the one that I'm particularly focused on is the beta isoform, which is encoded by PIK3CB. So what do we know about this right now? We know that pan inhibitors have been used, but there are many adverse effects associated. Selective inhibitors have been used in other cancers. For example, a delta inhibitor can be used in treatment of CML. We know for glioblastoma, the PIK3CA and the PIK3CB genes are more highly expressed. And previous research in Dr. Sheng's lab has shown that PIK3CB is actually a selective biomarker for glioblastoma recurrence and is the only one that correlates with higher rate, risk, and poor prognosis of glioblastoma patients, which is why we've decided to focus on it here. Additionally, we know that in normal cells, when we block the beta isoform, this was shown to be a little bit less toxic when compared to blocking the alpha isoform. So the gap in knowledge here is we still don't know what these isoforms, what roles they play in glioblastoma. We also don't know if we target specific isoforms, are we going to be able to cause greater treatment efficacy and produce less adverse effects as a result? So my specific research question is, does PIK3CB selectively regulate glioblastoma progression? We looked at this question through 4 different main domains, looking at first glioblastoma cell lines, glioblastoma patient samples gathered from Carilion Clinic, primary glioblastoma stem cells, as well as mouse models. Now, I did this project in conjunction with a PhD student, now a postdoc at VTCRI or FBRI, and my particular focus on the project was on the first 2, and he did most of the other half. So our specific hypothesis was that if we inhibit PIK3CB but not the other isoforms, we'll be able to selectively suppress the viability of glioblastoma cells that highly express this beta isoform. And at the same time, we may be able to have less toxicity to normal human astrocytes. The methods that I used to explore this question first included knockdown using short hairpin RNAs. This is a method of RNA interference. Secondly, we used PI3K isoform selective inhibitors to apply directly to glioblastoma cells and see the effect. So the results that you see here is the results of the first short hairpin RNA experiment. So short hairpin RNA is a way of RNA interference, basically allows us to silence certain genes and measure what kind of effect that has. So to walk you through it here, when we silence the PIK3CB gene, we get diminishing intensity of this band, indicating that PIK3CB, silencing this gene, inhibited downstream AKT activity, or it turned off that pathway. At the same time, when we silence this gene, it activates apoptosis, measured by these thickened bands here, indicating that we have more cell death. On the opposite end, when we silence PIK3CA, this actually had no effect at all. And so this led us to conclude that PIK3CB is actually more important for the survival of glioblastoma cells than the other isoforms. So my next couple studies, actually is the majority of my research, is looking at glioblastoma cell lines and then administering different selective inhibitors to see the effect that had on cell growth. So these are 4 of the major cell lines that I used: U87MG, SF295, A172, and LN229. Now, all of these had varying expression levels of these different isoforms, and what I want to draw your attention to is that U87 and SF295 are the ones that have high expression of beta isoforms. Therefore, we expect that these will respond more significantly. So this is a culmination of a lot of my work over the past couple years, and it's a really busy slide, but I'm going to walk you through it. So first, just want to bring your attention to the 4 cell lines that we had just talked about. When you look at the top third, these 2 figures here, we use 2 different beta inhibitors, GSK and TGX, and applied them to the cells. In the middle third, we use alpha inhibitors, 2 of them, called PIK75 and BYL719. And in the bottom third, we use 2 different delta inhibitors. So what's profound about these results is that when we applied these drugs in different concentrations from 0 to 25, we measured, so these drug treatments were given over a time period of 4 days, and then we measured the viability or the survival of these cells using something called an MTS assay. The MTS assay looks at the activity of a mitochondrial enzyme, which we expect to be alive and working if we have living cells. And so what was significant about this is that only the cells that had high expression levels of the beta isoform were selectively suppressed when treated with the beta inhibitors, which is exactly what we had expected. Interestingly, though, the alpha inhibitors and delta inhibitors had no such effect. In fact, they either killed all cells or had some killing in any random pattern that wasn't useful to us. Over here on the right-hand side, we have the IC50 values. So these are the concentrations of drug needed in order to induce 50% of killing of the cells. And what you notice here is that for the beta inhibitors, they actually really didn't require very high concentrations of doses in order to instill killing to these cells. Next, we wanted to see whether we could replicate our results in glioblastoma primary cells. So we gathered samples from the help of Dr. Simmons over at Carilion Clinic and cultured glioblastoma primary cells. We applied the similar inhibitors, and this figure basically confirms everything that we had in our previous results, and that the VTC103, which had high expression levels of the beta isoform, responded more significantly to the beta inhibitors. A little bit more information on the glioblastoma samples that we got from patients. We had a total of 8 of them, and actually 3 of the 8 had high expression levels of the beta isoform. What's really interesting about this data, though, is that when you look at the number of days till death, these patients had a significantly smaller amount of days to death compared to other patients who had low expression of this beta isoform, indicating that the higher expression of the beta isoform that you have, the more severe of a disease that you would have. So I talked a little bit about the MTS assay, which looked at mitochondrial activity and how we use that as a model of our measure of survival. So basically, we wanted to confirm that we are actually having cell death and not just inhibiting the activity of these mitochondrial enzymes. So we did something called a trypan blue exclusion assay. This basically was consistent with our MTS results. The next thing that we looked at was looking at animal models. So we did a mouse experiment using U87mg xenografts. So remember, the U87mg cells were the ones that had high expression levels of the beta isoform. So what we did is we injected these cells into the flanks of mice and then administered these beta isoform selective inhibitors, the TGX and the GSK, for a period of 2 weeks, and then measured the tumor volume every single day. And what you see here is that only the TGX was the only beta inhibitor to cause a significant reduction in tumor volume compared to control mice. So up to this point, we talked about how beta inhibitors are effective in not only glioblastoma cells, but also primary cells or patient samples, and also in animal models. But now the missing piece of the puzzle is how do these inhibitors affect normal brain tissue? So we looked at normal human astrocytes, and we applied these different inhibitors. So we have beta, alpha, delta, and some pan inhibitors. And we calculated the IC50 values over a treatment of 4 days. And what you notice is that the beta inhibitors required very high doses before we started to see killing of these astrocytes over this period of time. So this led us to conclude that PIK3CB inhibitors were actually way less toxic to astrocytes compared to the other forms of inhibitors. Now, if you remember a couple slides back, we had talked about how for these U87 and SF295 cells, they actually really didn't require a very high dose in order to have a killing effect. And if you recall back to pharmacology, if you have a drug with a low therapeutic effect, a low dose where you have a therapeutic effect, but a very high dose when you start to have a toxic effect, this results in a wide therapeutic index, which is a really great quality to have when you're looking about new drugs for treatment options for patients. So in conclusion, we have PIK3CB or P110 beta inhibitors are able to selectively suppress the survival of glioblastoma cells that have a high expression of this beta isoform. They also slow tumor growth in animal models and also have minimal toxicity to normal brain cells. Why is this important? Well, taking it back to precision medicine, where therapy is determined by tumor type, we believe that glioblastoma, or patients with this high expression value of the beta isoform, may be able to respond more significantly to PIK3CB or P110 inhibitors. And this may result in a new and potent selective target for a subset of glioblastoma patients. We believe this to be a tremendous step forward in development of new therapies for this really deadly disease. And with that, I'd like to thank you and thank my wonderful research mentor, Dr. Shang, as well as the rest of the team, as well as Lauren Harris, Dr. LeConte, and Dr. Vaughn for all of their support. Thank you.
Questions?
Cliff.
So, do you have any information about the patient characteristics besides time to mortality, about the human sample degrees?
That's a good question. I'm not aware of it at the moment. That was more, that figure was more of like a supplementary thing that we kind of just decided to look into. So we don't have that much data, but it's definitely something that is worth looking into if we want to look more into the clinical characteristics of those patients. But I'm not sure what we have exactly. Thank you.
Andrew Gaddis, who's next up on our list, worked with Dr. Cassandra Miersch of the Department of Orthopedics, conducting a randomized clinical trial looking at the amount of opioid prescription and how it affects use over time.
Thanks. All right. Thanks, Dr. LeConte. All right. Good afternoon, everybody. I know it's late in the day. I hope everyone's sufficiently caffeinated. And on that note, hoping you're interested in some work that I've done with my really fantastic mentor, Dr. Cassandra Miersch from Orthopedics. The title of our study was called The Effect of Initial Prescription Size on Opioid Use and Patient Satisfaction After Minor Hand Surgery, a Randomized Controlled Trial. It's a bit of a mouthful, but again, hoping you're interested in it. Before we get started, I just wanted to thank some of our co-authors. They put like endless time into this study, and I think it wouldn't have been possible without them, so I wanted to acknowledge their efforts up front. So to provide some context into our research study, I think it's important to get a scope of what was going on in terms of opioid prescribing back in 2014 when this study was conceptualized and designed. And so as you can see from this map, this is data from 2012, and this shows the number of opioid prescriptions in each state. And so for each of these states that's outlined in purple here, you can see that there's, in a calendar year, more opioid prescriptions in those states than people living in them in the first place. So we have this historical and ongoing opioid prescription problem. And where we are in Southwest Virginia, you can see where we're surrounded by these states that are outlined in purple. So it's a particularly relevant issue for this region. So if you look at the literature that's out there on opioids, and there's quite a bit of it, most of it focuses on kind of long-term exposure to opioids for chronic pain. So this graph illustrates the concept of tolerance, right? So, you know, people who are on opioids in the long term, they tend to require the same amount of kind of opioid and then an ever-increasing amount of opioid to meet that same analgesic effect. There's also the concept of opioid-induced hyperanalgesia, which basically states that the longer that you're on opioids, your pain threshold is reduced, and so you're more susceptible rather than less susceptible to chronic pain. So in general and in summary, we are fairly confident that opioids, at least for long-term and chronic pain, are a poor intervention. But what we don't know as much about is the acute exposure to opioids. This is some data from an animal model, and this is data showing that, at least suggesting that tolerance to opioids, rather than happening in the long term, can happen at least within weeks, if not within days. So our research team feels like this acute exposure to opioids is an under-researched topic and an area that definitely warrants some studies. And so when we're talking kind of about what's out there, we're stuck with more associational and observational data. And this is a table from a study that looked at claims data, so looking at insurance claims across the entire nation. And so we can look at some minor procedures. So they looked at minor outpatient upper extremity procedures. And these are, you know, again, outpatient procedures not expected to cause chronic reliance upon opioids. But what you can see here is that the average rates are pretty alarming, even for these minor procedures. So these are opioid naive patients going into these surgeries, not having other procedures, And then down the line, we're finding on average it's about 13.5%, between 10 and 15% overall. For these minor procedures of these patients wind up chronically prescribed opioids. This again illustrates our concern that this acute exposure to opioids does represent a risk for the patient. And kind of the landmark study that we felt like really spurred us to create our study was this 2012 study, which basically just tracked patients after these minor outpatient upper extremity procedures, and they wanted to know how many opioids are patients actually using. And so you can see here that 77% of their patient population from this clinic took less than half of the number of opioids that were prescribed to them. This is a risk not only to patients, but this is also a risk to the general public. This is nationwide survey data done by SAMHSA. And so the big graph here in blue, you can see that this is people getting illicit or kind of non-medical opioids, and most of them are getting them from a friend or family member. And then if you look at the smaller graph here, if you break that out, most of those friends or family members are getting them from a physician directly. So as a medical community, this is a problem that we're directly contributing to. So the gap in knowledge that we set out to address is that at the time we designed this study, there really was no existing evidence regarding the effects of initial post-surgical opioid prescription size on post-surgical opioid consumption. And our hypothesis is that this initial prescription size is a modifiable factor, and that smaller initial prescription size might not only reduce the number of unused opioids available for diversion, but also might influence opiate consumption itself, with patients showing a reduced number of opioids consumed in the first place. And then finally, that this might not have much of an effect at all on patient satisfaction. So in order to set our study up, we relied on some legacy data from this institution. We looked at 2 very commonly performed outpatient upper extremity procedures, and we found that on average, patients after these procedures were given 40 to 55 milligram hydrocodone equivalents. And so based on that, we designed our study. So we decided to use a randomized controlled trial, prospective in nature. Our intervention had 2 wings, so we developed a small initial prescription size group of 10 pills, and then also a much larger initial prescription size group of 30 pills. I think it's worthwhile to note that even our large initial prescription size group of 30 pills represents a decrease from the historical prescribing practices at the institution. In terms of patient inclusion, we included patients aged 19 to 69. Important consideration, we only included patients that were not taking opioid at the time of surgery, so we excluded patients that weren't opioid naive. We also did not include patients that had allergies or major surgical complications, and we took care to select a group of procedures that we felt were very similar. So these are minor surgeries in the soft tissue or minor bone. They're in the same general anatomical location of the hand or the wrist, and they're relatively commonly performed and expected to have similar post-surgical pain requirements. We used a computer to generate 200 randomly sealed enrollment packets. We distribute them to 3 sites, surgical sites, again, kind of randomized. In terms of enrollment, our surgeon investigators obtained consent from all of our patients for the study. And so our investigators and our patients were blinded to both treatment groups before consenting the patient and opening the packet. And then upon opening the packet, the patient was blinded to the other intervention group. So they only knew how many pills they would get. They wouldn't know how many pills anyone else in the study would get. In terms of recommendations for post-surgical pain, our surgeon investigators followed these FDA guidelines, which basically says 1 to 2 pills every 4 to 6 hours, a max of 8 pills per day. Only take them if you need them. And I think it's really important to stress that refills were not limited by our study protocol. So when patients were considering joining the study, they were told that, look, if you're still in pain after your refill is out, please contact your physician. And our surgeon investigators were told to give refills as they saw fit. In terms of data collection, we used a written form at each patient's intake, which recorded characteristics about the patient and also recorded characteristics about the surgery that was performed. And then 10 to 14 days later, we did a phone interview, which surveyed the patient about how the surgery had gone, how their satisfaction was in terms of pain, how many pills had they taken, how many were left over, et cetera. And then last, we used retrospective chart review, both to generate the preliminary data for the study and then also just to monitor allocation. These are our primary outcomes. So our primary outcome was opiate consumption. We just wanted to know how many pills are people taking in each group, and then how many do they have left over. Secondary outcomes included the number of refill requests that would be made by each patient, days on narcotic medication, whether the patient was still taking narcotic at follow-up, was their satisfaction with their surgery and their pain control, and is it comparable between groups. And then last, what their opinion was regarding the prescription size that they were given. We collected these on different scales. So in terms of efficacy of pain control, we used a 4-point scale ranging from poor to excellent. In terms of satisfaction with both pain and with their surgery, we rated on a 4-point scale between extremely dissatisfied and extremely satisfied. And then last, in terms of the appropriateness of prescription, we used a 3-point scale ranging from too few to too many. And this is our consort enrollment diagram. So we screened 544 surgical candidates for this study. We wound up enrolling 200 of them. I think it's worthwhile to note that we excluded 136 patients based on whether or not they were using narcotics in the preoperative period. So again, an important area for follow-up study. But we enrolled 200 people successfully in this study, randomized them, and we wound up with 176 after exclusions. So we were happy with our sample size. In terms of our randomization, there were no significant difference between both groups in age, gender, anesthesia type, aspects of surgery. We felt overall it was a very successful randomization. We also noted that there was no difference in opiate consumption between procedure types, which we felt was an indicator that the surgeon investigators that we worked with put together a good list of procedures that were very similar in nature. And so these are our key results. So kind of dwell on this for a minute here. So picture on the left here is our smaller initial prescription size group of 10 pills. Picture on the right is our larger initial prescription size group of 30 pills. And so from left to right, we're looking at the number of pills prescribed, and then the number of pills used here, and then the number of pills left over here. And so our results are fairly significant. So for our larger or 30 pill initial prescription size group, they used about 12 pills, which is about twice as many as our smaller group of 10 pills. They also had about 4-fold medication left over. So they had on average about 20 pills left over compared to roughly 5 from our smaller initial prescription size group of 10 pills. So again, very significant differences in terms of opiate consumption and number of opiates left over between these two groups. Some other findings we found to be very much of interest is that our 10 pill or our smaller initial prescription size group tended to initiate narcotic use less than the other group. Very worthwhile to note, our refill request was the exact same. So each group requested refills at a rate of 14%. So physicians that might be writing larger prescriptions to avoid phone calls for refills, that just doesn't seem to be founded in the facts found by our study. In terms of days on narcotic, interestingly, both groups had the same number of days on narcotic for patients who discontinued narcotic use before the follow-up phone call. So it seems that in each day, the patients with the larger initial prescription size group are just taking more. And very importantly for us, even though our numbers are very small, our larger or 30 pill initial prescription size group was much more likely to be still taking narcotic at follow-up. And so we're wondering if this initial prescription size might be a risk factor for chronic opioid use. In terms of satisfaction and efficacy of pain control, again, a 4-point scale, and where we really want our patients to live is kind of in these two blue columns. And so you can see from this diagram that most of our patients, in terms of efficacy of pain control, rated it as good or excellent, which is where we want them to be. There were no statistical differences in the distribution between the two groups. In terms of satisfaction with pain control, we found the exact same thing. Most of our patients were satisfied or extremely satisfied with their pain control, regardless of how many pills they received. No statistical differences between these two groups. In terms of their experience with their surgery overall, most patients were satisfied or extremely satisfied with their surgery, regardless of what group they were placed in. No statistically significant differences between groups. And this is a really interesting finding. So we asked them how they felt about the prescription that they got. And so you can see in our 10 pill group, the majority of our patients in this dark kind of blue line here, they felt that their prescription was about right. So it seems like 10 pills is the expectation from patients. And you can see in our 30 pill group, most of the patients felt that this was too many pills. So it seems like this 30 pill prescription size is just not in line with patient expectations in the first place. So in summary, our group that received smaller opioid prescriptions we found that they used decreased number of pills in general. They had a decreased number of unused pills left over, did not increase refill requests, did not change patient satisfaction. This discouraged prolonged opioid use. It discouraged the initiation of opioid medication in the first place. And also just in general, we're better in line with patient expectations. While we were doing this study, some supportive literature came out. We're of course not the only group looking at this, even though I will stress that all of their data is observational or associational. So this is a point of great concern for Medicare and Medicaid, and so the Center for Medicare and Medicaid came out with this study, which basically shows that initial prescription size is related in a linear fashion to the percentage of chance that that patient will wind up chronically prescribed opioids, and these are also for opioid-naive patients. So again, associational data, but it seems like our conclusion is supported by nationwide data. In a similar fashion, this is a group looking at opioid consumption post-cesarean section. And again, this is associational data, even though it is prospective. And so what they found is similar to our findings. They found that patients who received more opioid prescription, kind of larger initial opioid sizes, wound up consuming more opioid pills. So what's going on here? Why do we see these findings? And I think one way to explain it could potentially be the anchoring bias. And this is a psychological concept where people tend to focus very much on the first piece of information given to them when solving a problem. And so the example shown here is that if you ask a group of people to predict someone's lifespan, and you give them a larger number in the question prompt, the average answer is going to be larger. And if you ask the same question and you give a smaller number in the question prompt, the average answer will be lower. So what we're thinking is patients are kind of fixating on this initial prescription given to them, and whether they stray from that a little bit on either end, they're still feeling like this is the number of pills that's going to help them get over their post-surgical pain. In a related vein, we suspect that there's a lot of nonverbal communication going on here. So as students, we know we're always talking about, maintaining eye contact and posture and all the things listed here. It's very important for generating the patient outcomes that you want. And so we suspect that outside of just body language, the prescription itself represents some form of nonverbal communication, right? So when patients get this prescription, they're hearing that this is a prescription size that their physician thinks is reasonable and safe, and so therefore that might influence how they take it. Some other findings that we figured we would report would be there were older patients tended to take less opioids in general, and that's pretty well supported in the literature. Also, type of anesthesia showed us a significant correlation with opioid use, with our patients receiving local anesthesia, taking the fewest number of opioid pills. And this is also supported with the literature, but we find it particularly interesting in our study because the procedures that we selected for this project were procedures where the patient basically, or they can be performed safely with both general and local. And so we thought it was interesting in that respect and probably worthy of follow-up. In terms of limitations, we only included opioid-naive patients. This was a pretty significant limitation. We know that preoperative opioid use is a good predictor of postoperative opioid use, so hopefully future studies will include those patients. We did not control for, although we of course encouraged the use of non-opioid co-analgesics, so things like NSAIDs of course are a valuable piece of pain control, and hopefully in the future studies will standardize for those so we could better isolate the effect of opioids. We are restricted to a very small anatomical location and a small single center study in a single region. So of course we could generalize these findings, and we're hoping to reach out to surgeon collaborators in different disciplines and kind of expand this study and potentially kind of other future directions I guess would be to use other multi-center data such as statewide data, claims data, et cetera. Despite the limitations, we were successfully published in Annals of Surgery just recently, and we're really proud of this. It's a fairly highly cited surgical journal, so again, we're hoping that this will lead to kind of future studies. And again, as I had said, some of these future directions we're hoping to continue to pursue. We have pursued on a very preliminary basis one of them, which is to look at claims data from the state of Virginia. So we looked at this database called the APCD, or the All Payers Claims Database. So we looked at statewide prescriptions for a couple of years, and we can see that the same findings. So we found that initial prescription days supply for the first prescription is correlated very much and in a linear fashion with the rate of conversion to chronic opioid therapy. There's tons of people to acknowledge, everyone involved, especially the patients who participated. But yeah, kind of could go on for a while, but happy to answer questions if anyone has any. Thank you for your time.
You mentioned that you're thinking about looking at substitution of other analgesics in the future. Do you have any data from the exit interviews of patients on what amounts of other analgesia they use after the procedures you've broken?
Sure, that's a really important aspect of this study, and so it's not something we included in the initial design. As the study progressed, we thought about potentially adding that to the questionnaire, but to be honest, because we didn't include it in the initial study design, we felt that kind of getting into it later would just open up the study to weaknesses that we weren't really prepared to deal with. So that's definitely something that in the future, though, we're hoping to include in any sort of future iteration of this study. But great question.
What do you think in terms of, do you think there's an effect on the patient's level of understanding of the opioid epidemic or their education level? Do you think that would have a significant impact on the results of the level of satisfaction? Because obviously you're looking at a population of people that might be more highly consumed. No wonder that difference would compare to state.
Yeah, that's a really good question. And it's interesting because throughout the course of this study, the opioid epidemic and the public awareness campaigns around it kind of happened, right? So that kind of changed throughout the course of the study. I think really that kind of gets back to our exclusion of patients already taking opioid at the time of surgery. You know, that's why I think that that's a particularly valuable area of future study, because, you know, I think that this is very applicable and well designed for people who aren't taking opioids at the time of surgery, but, you know, that's not every patient, and there's quite a few patients who are already on opioids when you see them for a surgical consultation. And, you know, I think that for those folks, I'm not sure these same principles would apply. So I think that that's a group of people who are, you know, obviously very vulnerable, and a group of people that, you know, specific studies should be targeted on in the future. Also, I think that, you know, provider education around the opioid epidemic, I bet also has a very significant factor in terms of opioid prescriptions, and that is probably also something to look into. But yeah, great thoughts, thanks.
And last of the day, but certainly not least, is David Barton. So David worked with Dr. Jonathan Carmouche, also of the Department of Orthopedics. David will first describe his research that established the need for improved management of patients at risk for fractures due to osteoporosis, and then describe the implementation of an intervention designed to target that need.
Thank you, Dr. Vaughn. So without further ado, no disclosures. I'm going to go through a little bit of general background, then talk about the different phases of this study, and discuss each in turn as we go. So fragility fractures are classically defined as a fracture occurring after a fall from standing or other low energy mechanism. They're also known as minimal trauma fractures and are classically associated with low bone mass, especially osteoporosis. Fragility fractures are extremely common, with 1 in 2 women and 1 in 5 men having a fragility fracture at some point in their lives. And that comes out to more than 2 million fractures a year in the United States, with substantial associated healthcare costs and morbidity and mortality. Having any fracture doubles your risk of future fractures, and FDA-approved medications can reduce that risk by 60 to 80%. Given that need and those statistics, it makes sense that a lot of fracture prevention efforts have focused on catching patients after an initial fracture and trying to prevent future fractures. Unfortunately, we're still really not doing a very good job of that. In the classical osteoporosis population, with Medicare data, less than 40% of patients received screening or treatment for osteoporosis in the year following a fracture. Given that national treatment gap, we decided to investigate how well are we doing at managing those patients at Carilion Clinic. Therefore, we undertook a retrospective cohort review, including patients aged 50 years of age and older, who presented to the emergency department for an ICD-9 code of a classical osteoporosis fracture type. We only included their first fracture of a particular type, and we went from EPIC implementation in July 2008 through September 2014, which was exactly 2 years before the ICD-9 to 10 crossover, in order to allow for consistent coding. We were interested in anti-osteoporosis prescribing rates, vitamin D and calcium supplementation, rates of DEXA scanning, and rates of new fractures. We had almost 3,000 vertebral fracture patients included in the study, more than 1,000 hip fracture patients, almost 1,000 pelvic fractures, and more than 1,500 humerus fractures. Average ages were in the mid-70s, with a female predominance, as is consistent with osteoporosis in general. We investigated the frequency of high-energy mechanisms through 2 different ways. One is ICD-9 and 10 codes actually allow for coding of specific trauma mechanisms. Unfortunately, 0 of the fractures included in this study had a mechanism coded into them during the emergency department visit. So we looked at mechanism indirectly by looking at the frequency of other fractures that were documented during the same emergency department visit. And we found that that rate varied between 5 and 11% depending on the fracture type. We looked at DEXA scanning rate as a surrogate measure for osteoporosis evaluation, given that the World Health Organization guidelines on osteoporosis say that a DEXA scan is necessary in order to officially diagnose osteoporosis. We found that while about 2% of those patients had had a DEXA scan in the previous 2 years that could be used for diagnosis, less than 1% were receiving a new DEXA scan to formally evaluate them for osteoporosis in the year following fracture. We really would like to see that
be almost all of the remaining patients. Similarly, for prescription management of osteoporosis, we found that while about 20% of patients were on an osteoporosis medication before their fall, only about 5%, depending on fracture type, received a new medication in the year following fracture. And again, most of these patients met criteria for starting an osteoporosis medication. So these really go all the way across if we were doing this right. If we're generous and include calcium and vitamin D supplementation in this, calcium and vitamin D supplementation generally being a necessary but not sufficient method of treatment that is often used first by people who are less comfortable with osteoporosis medications, we find that while the rate of treatment is slightly higher, we're still neglecting more than 50% of patients who are not receiving any treatment at any time point we investigated, not just in the first year after fracture. Furthermore, we found that the rate of treatment actually decreased over time during our study period for vertebral fracture patients in a statistically significant manner, and we found the same trend, although it was not statistically significant, for pelvic fracture patients. This has actually been demonstrated elsewhere in the literature and is thought to be associated with excessive fear of atypical femur fractures and osteonecrosis of the jaw, which, while they do occur in this patient population, are much less common than they are in at chemotherapy doses for these medications, and generally get overhyped, and patients and providers who are not expert in this area tend to overemphasize those. And to underscore the importance of this, I wanted to point out how many of these patients went on to have an additional fracture within 2 years. Almost 40% by fracture type. So a lot of those fractures could be prevented if we were doing a better job of managing these patients. So given we've established the osteoporosis treatment gap at our institution, we went on to look at 2 different ways to address that gap, either a protocol-based approach or a fracture liaison service. Fracture liaison services, for those who are not familiar, are an emerging gold standard for how to evaluate patients for osteoporosis and manage them post-fracture. There's generally a diffusion of responsibility in this area with no single provider taking ownership. Fragility fracture liaison services solve that by having a dedicated person whose job it is to screen and treat patients for osteoporosis after a fracture. We sent out 2 surveys to the Department of Orthopedic Surgery, 1.5 years apart, looking at physician buy-in and barriers to implementation for these 2 different methods. We got a 77% response rate on the first survey and required participation on the second one. So one of the things that came out of this was that we found that overwhelmingly orthopedic providers recognize that we're not doing a completely adequate job of osteoporosis management in either the primary care or orthopedic setting after a fracture, with only one provider thinking we are. Respondents' perceived responsibility. So most orthopedic surgeons felt moderately responsible for evaluating osteoporosis after fracture. They were generally fairly comfortable with diet and exercise and fall avoidance counseling, but we're not very comfortable with medication side effects or drug counseling. Red here being uncomfortable, green being comfortable. I generally keep with green being good, stoplight themes here. So steps that respondents report taking in managing osteoporosis. While a number of providers reported being relatively good about ordering DEXA scans, vitamin D levels, and referring patients for osteoporosis assessment, not as good as we would want, but they're doing some of it. We found that almost none of them are initiating pharmacological management for these patients, which is really what they need. And we found, once again, that while they're comfortable and are using calcium and vitamin D supplementation, almost none of them are using the actual FDA-approved medications for osteoporosis because they report being uncomfortable with them. And the reasons, as I alluded to before, first and foremost, they report limited experience with this. These respondents identify primarily as surgeons rather than as medical doctors, and therefore are less comfortable with the pharmacological aspect than they are with the surgical aspect management of these patients. They also express the excessive concern that I alluded to before about osteonecrosis of the jaw and atypical femur fractures. And then, given all these results, it's not surprising that they strongly preferred to have a fracture liaison service versus a protocol-based approach. So given the preference for fracture liaison service, we went and did a systematic literature review and meta-analysis looking at how effective fracture liaison services are on the clinical outcomes we care about, that being DEXA scanning, the rate of new medication prescribing, the rate of new fractures, and mortality. We only included the most rigorous studies that were available on this with a strong comparator group from a similar institution or pre-post at the same institution. We included 10 studies in our qualitative analysis and 5 in our quantitative analysis. We found that DEXA scanning rates improved with FLS implementation, and anti-resorptive prescribing rates also improved. We found at various time points time-dependent effects on the rate of new fractures and the rate of mortality following implementation. This all led to us opening a fracture liaison service at Carilion Clinic in October 2017. This is not actually from this. So Lisa Wright runs our fracture liaison service. It's located in Riverside 3 in the orthopedic trauma group so that they can have same-day appointments with the fracture liaison service at the same time they're coming to orthopedic follow-up. And between October 2017 and December 2018, we had 890 visits in that center, which came to about 23% of our theoretical capacity, or probably about 50% of what we can actually see in that clinic. And that came out to 600 unique patients. This can be accessed in the outpatient order set for Carilion Clinic under FLS, bone health, or fracture liaison, using those as your search terms. Please send referrals. Our referrals so far have come predominantly from orthopedics. As you can see, 86% of patient referrals came from orthopedics, with orthopedic trauma and orthopedic spine, which are our two physician thought leaders on this, accounting for the predominant referral sources, although family medicine has also contributed a significant number of cases, and we've even gotten a couple referrals from endocrinology and hematology oncology. That came out to about 3 new referrals per day. And we sent more than 400 patients for some sort of bone mineral density testing out of that clinic. Started over 180 patients on a biologic for osteoporosis, either Forteo, Tymlos, or Prolia. Sent 27 patients for IV bisphosphonates. Sent 57 for fall prevention. And we actually found more than 50 patients who had suspected MGUS or other hematological abnormalities and have been sending those to HemOnc for that. And we've also found a number of cases, almost 30 cases of hyperparathyroidism, which we've sent out for referral as well. So I did a preliminary data analysis. This center's only been open for a little over a year, so the data follow-up is not as rigorous as we would like, but we did some preliminary analysis here, and I'll show you what I have. So we chose vertebral fractures because they're our largest fracture group. We had 2,933 vertebral fractures in our retrospective cohort, which came out to about 450 per year, or 290 in the if we're going to parallel the 8-month increment that we had of patients, since we used 8 months of patient data with 6 months of follow-up for our analysis. So our power analysis suggested, which we ran based on 1 year of data, which suggested that we needed 400 patients, and we would need 38% of them, or 38 of them to receive treatment and 12 to receive a DEXA scan in order to be able to demonstrate a difference here. We also saw that in order to show a difference in fractures, not only would we need 2 years of follow-up, but we would need to drop the refracture rate from 38% to 31%, which really was not feasible given the time constraints in this project. What we actually observed was in the 8 months we included, we had 509 vertebral fracture patients, of whom 18 received a osteoporosis medication and only 2 received a DEXA scan. This comes in part because a lot of the patients that were seen in the clinic were actually patients who had not had a fracture, and we're not necessarily capturing all of the fracture patients that we were hoping to see. We have not seen a statistically significant change in prescribing rates for treatment-naive patients for either vertebral fractures or hip fractures. So next steps for this are to rerun the analysis with more data going forward. We were initially planning to do 1 year of full patient visits with 1 year of follow-up. And it would be even better if we could do 2 years of patient visits to allow the center to mature more before we're really passing judgment on it with a full year of follow-up. As well as eventually I'd like to see refracture data to see whether we've been able to successfully reduce the rate of new fractures, because that has not been shown very well in the United States. We'd also like to look at different ways to increase the FLS referral rate by looking at factors that influence who gets a referral for fracture liaison service consult and who actually shows up to those consultations. And then any interventions that we could put in place to increase that referral rate. I think there's also an interesting niche here in showing how, in discussing the role of the FLS in the detection of MGUS and hyperparathyroidism. That's really not something we had expected when we implemented the center. And it's an area that really hasn't been discussed in the literature. So I think that is warrants a study of its own. And then because we now have a dedicated osteoporosis treatment center in the Department of Orthopedics, there's a role for that center to be involved in clinical trials on osteoporosis, both randomized multi-centered trials and preliminary trials that are started out of here. Questions? Cliff?
So I noticed, I noticed that the rate of refracture in vertebral fractures was substantially higher in the retrospective target view than for other fragility fractures. And I was wondering, is there any data in terms of what treatments those patients receive? For example, on kyphoplasty in the chart view that's done.
So I do have data on procedures that were performed on those patients. I haven't dug into it as to whether they had kyphoplasties or not. That would be an interesting follow-up study. Zainab?
You mentioned there's some ambiguity around who's responsible for taking care of patients that have sustained fragility fractures.
Yes.
What kind of non-orthopedic involvement do you have with the fracture liaison service, or is it completely just orthopedics?
So it is run by orthopedics at this point. There was a lot of horse trading in order to get that set up, a lot of negotiations with internal medicine, et cetera. And our nurse practitioner is actually trained in family medicine as opposed to being trained in orthopedics. But it is entirely run by orthopedics at this time.